electronics, eco-friendly transportation, advanced materials and nanotechnology research, social sciences and humanities, consisting of people and population, culture and language, policy and governance or good governance, local government and regional autonomy, social conflict, economics and poverty research, scientific services, consisting of scientific repository, metrology and standardization, innovation of technology transfer and utilization of science and technology services. Studies into scientific and technological developments and scientific community training. LIPI researchers are supported by world-class facilities spread over 48 research centers and technical implementation units throughout Indonesia. LIPI also has research vessels for oceanography research activities, as well as other marine research expeditions. We understand the importance of maintaining the natural ecosystem and its contents. To that end, LIPI has collections of flora and fauna from all over Indonesia collected and recultivated in its 5 botanical gardens: Bogor Botanical Garden, Cibodas Botanical Garden, Purwodadi Botanical Gardens, Bali Botanical Gardens, and Cibinong Botanical Garden. LIPI manages world-class flora-fauna collections. The Bogorienza Herbarium Museum, established in 1817, is the 3rd largest herbarium museum in the world. The museum has the 10th largest reference collection of fauna specimens in the world, and Indonesia Culture Collection, or INA-CC, which holds LIPI's collection of microorganisms listed in the World Microorganism Collection Culture Federation. Given its vast resources, LIPI is also appointed as a scientific authority in 1. Guiding, training, assessing, and authorizing national researchers, 2. Accrediting scientific journals, 3. Studying and evaluating biodiversity, establishing botanical gardens, as well as scientific testing and measurement. LIPI also plays a strategic role in various international scientific activities. This is evidenced by LIPI occupying the trusted position as the international focal point for UNESCO. quality of life of Indonesians, as well as the development of science in Indonesia. To encourage the spirit of research among the younger generation, LIPI has, since 1968, hosted research paper competitions for teenagers, as well as the National Young Inventor Award, which is keenly followed by students throughout Indonesia. We are ready to cooperate with various elements, both national and international, to realize our vision of becoming a world-class research institution to improve the competitiveness of Indonesia in the future. Each vision begins with a spark of inspiration, and each dream is driven by the passion to realize it. For over 50 years, we have worked hard to serve Indonesia. Becoming a world-class research institution that uses research to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences, or LIPI. LIPI was formally established in 1967, but its history goes back as far as 1817, with the establishment of a plant conservation center at the Bogor Botanical Garden. We are spread over 11 provinces. We have over 4,800 employees. We are the largest research institute and the largest patent contributor in Indonesia. LIPI is directly responsible to the President of the Republic of Indonesia with the coordination of results for Indonesia. LIPI's research focus areas include life sciences, consisting of molecular biology, biotechnology and medicine, biomedical science, molecular and post-genomic agriculture, exploration and utilization of biodiversity research, earth sciences, consisting of marine biology and inland waterways, eco-hydrology, coastal and marine areas, climate change and disaster management, and mineral resources research. Engineering sciences, consisting of defense and IT security, telecommunications and electronics, eco-friendly transportation, advanced materials and nanotechnology research. Social sciences and humanities, consisting of people and population, culture and language, policy and governance, or good governance, local government and regional autonomy, social conflict, economics and poverty research, scientific services, consisting of scientific repository, metrology and standardization, innovation of technology transfer and utilization of science and technology services. Studies into scientific and technological developments and scientific community training. LIPI researchers are supported by world-class facilities spread over 48 research centers and technical implementation units throughout Indonesia. LIPI also has research vessels for oceanography research activities, as well as other marine research expeditions. We understand the importance of maintaining the natural ecosystem and its contents. To that end, LIPI has collections of flora and fauna from all over Indonesia collected and recultivated in its 5 botanical gardens. Bogor Botanical Garden, Cibodas Botanical Garden, Purwodadi Botanical Gardens, Bali Botanical Gardens, and Cibinong Botanical Garden. LIPI manages world-class flora-fauna collections. The Bogoriense Herbarium Museum, established in 1817, is the 3rd largest herbarium museum in the world. The museum has the 10th largest reference collection of fauna specimens in the world, and Indonesia Culture Collection, or INA-CC, which holds LIPI's collection of microorganisms listed in the World Microorganism Collection Culture Federation. Given its vast resources, LIPI is also appointed as a scientific authority in 1. Guiding, training, assessing, and authorizing national researchers, 2. Accrediting scientific journals, 3. Studying and evaluating biodiversity, establishing botanical gardens, as well as scientific testing and measurement. LIPI also plays a strategic role in various international scientific activities. This is evidenced by LIPI occupying the trusted position as the international focal point International Center for Interdisciplinary and Advanced Research, ICR, and Marine Biodiversity and Ecosystem Health Center, or MARBEST Center, engaged in marine training. In addition, LIPI is also active in other international organizations, such as Science Council of Asia, SCA, Botanic Gardens Conservation International, BGCI, International Council for Science, ICSU, and others. LIPI works with the following stakeholders to disseminate its research results to the community, namely, national ministries and state-owned enterprises in the field of metrology and testing, local government, universities, and industries, including the private sector. Since 2010, more than 171 MOUs of cooperation have been established between LIPI and the national stakeholders. This cooperation has resulted in the establishment of various science and techno parks. These parks incubate technology startups and facilitate technology transfer. LIPI has also established more than 70 partnerships with international partners since 2010 in various branches of science across the world. Including research collaboration, workshops for teenagers, as well as the National Young Inventor Award, which is keenly followed by students throughout Indonesia. We are ready to cooperate with various elements, both national and international, to realize our vision of becoming a world-class research institution to improve the competitiveness of Indonesia in the future. Each vision begins with a spark of inspiration. And each dream is driven by the passion to realize it. For over 50 years, we have worked hard to serve Indonesia. Becoming a world-class research institution that uses research to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences, or LIPI. LIPI was formally established in 1967, but its history goes back as far as 1817, with the establishment of a plant conservation center at the Bogor Botanical Garden. We are spread over 11 provinces. We have over 4,800 employees. We are the largest research institute and the largest patent contributor in Indonesia. LIPI is directly responsible to the President of the Republic of Indonesia, with the coordination of the Ministry of Research, Technology, and Higher Education. LIPI's acting chairman is Professor Dr. Bambang Subianto, who is assisted by the Vice Chairman, General Secretary, and 5 deputies who each oversee research centers and technical service units in their respective fields. LIPI has a strong foundation built on conducting research that delivers large-scale, significant, and real results for Indonesia. LIPI's research focus areas include life sciences, consisting of molecular biology, biotechnology and medicine, biomedical science, molecular and post-genomic agriculture, exploration and utilization of biodiversity research, earth sciences, consisting of marine biology and inland waterways, eco-hydrology, coastal and marine areas, climate change and disaster management, and mineral resources research. Engineering sciences, consisting of defense and IT security, telecommunications and electronics, eco-friendly transportation, advanced materials and nanotechnology research. Social sciences and humanities, consisting of people and population, culture and language, policy and governance, or good governance, local government and regional autonomy, social conflict, economics and poverty research. Scientific services, consisting of scientific repository, metrology and standardization, innovation of technology transfer and utilization of science and technology services. Studies into scientific and technological developments and scientific community training. LIPI researchers are supported by world-class facilities spread over 48 research centers and technical implementation units throughout Indonesia. LIPI also has research vessels for oceanography research activities, as well as other marine research expeditions. We understand the importance of maintaining the natural ecosystem and its contents. To that end, LIPI has collections of flora and fauna from all over Indonesia collected and recultivated in its 5 botanical gardens: Bogor Botanical Garden, Cibodas Botanical Garden, Purwodadi Botanical Gardens, Bali Botanical Gardens, and Cibinong Botanical Garden. LIPI manages world-class flora-fauna collections. The Bogorienza Herbarium Museum, established in 1817, is the third largest herbarium museum in the world. The museum has the 10th largest reference collection of fauna specimens in the world, and Indonesia Culture Collection, or INA-CC, which holds LIPI's collection of microorganisms listed in the World Microorganism Collection Culture Federation. Given its vast resources, LIPI is also appointed as a scientific authority in 1. Guiding, training, assessing, and authorizing national researchers. 2. Accrediting scientific journals. 3. Studying and evaluating biodiversity. Establishing botanical gardens, as well as scientific testing and measurement. LIPI also plays a strategic role in various international scientific activities. This is evidenced by LIPI occupying the trusted position as the international focal point for UNESCO and has 3 international centers, namely Asia-Pacific Centers for Ecohydrology, APCE, International Center for Interdisciplinary and Advanced Research, ICR, and Marine Biodiversity and Ecosystem Health Center, or MARBEST Center, engaged in marine training. In addition, LIPI is also active in other international organizations, such as Science Council of Asia, SCA, Botanic Gardens Conservation International, BGCI, International Council for Science, ICSU, and others. LIPI works with the following stakeholders to disseminate its research results to the community, namely, national ministries and state-owned enterprises in the field of metrology and testing, local government, universities, and industries, including the private sector. Since 2010, more than 171 MOUs of cooperation have been established between LIPI and the national stakeholders. This cooperation has resulted in the establishment of various science and techno parks. These parks incubate technology startups and facilitate technology transfer. LIPI has also established more than 70 partnerships with international partners since 2010 in various branches of science across the world. Including research collaboration, workshops, and exchange programs and scholarships for LIPI researchers. For more than 50 years, LIPI has produced research that has significantly improved the quality of life of Indonesians, as well as the development of science in Indonesia. To encourage the spirit of research among the younger generation, LIPI has, since 1968, hosted research paper competitions for teenagers, as well as the National Young Inventor Award, which is keenly followed by students throughout Indonesia. We are ready to cooperate with various elements, both national and international, to realize our vision of becoming a world-class research institution to improve the competitiveness of Indonesia in the future. Each vision begins with a spark of inspiration. And each dream is driven by the passion to realize it. For over 50 years, we have worked hard to serve Indonesia. Becoming a world-class research institution that uses research to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences, or LIPI. LIPI was formally established in 1967, but its history goes back as far as 1817, with the establishment of a plant conservation center at the Bogor Botanical Garden. We are spread over 11 provinces. We have over 4,800 employees. We are the largest research institute and the largest patent contributor in Indonesia. LIPI is directly responsible to the President of the Republic of Indonesia, with the coordination of the Ministry of Research, Technology, and Higher Education. LIPI's acting chairman is Professor Dr. Bambang Subianto, who is assisted by the Vice Chairman, General Secretary, and 5 deputies who each oversee research centers and technical service units in their respective fields. LIPI has a strong foundation built on conducting research that delivers large-scale, significant, and real results for Indonesia. LIPI's research focus areas include life sciences, consisting of molecular biology, biotechnology and medicine, biomedical science, molecular and post-genomic agriculture, exploration and utilization of biodiversity research, earth sciences, consisting of marine biology and inland waterways, eco-hydrology, coastal and marine areas, climate change and disaster management, and mineral resources research. Engineering sciences, consisting of defense and IT security, telecommunications and electronics, eco-friendly transportation, advanced materials and nanotechnology research. Social sciences and humanities, consisting of people and population, culture and language, policy and governance, or good governance, local government and regional autonomy, social conflict, economics and poverty research. Scientific services, consisting of scientific repository, metrology and standardization, innovation of technology transfer, and utilization of science and technology services. Studies into scientific and technological developments and scientific community training. LIPI researchers are supported by world-class facilities spread over 48 research centers and technical implementation units throughout Indonesia. LIPI also has research vessels for oceanography research activities, as well as other marine research expeditions. We understand the importance of maintaining the natural ecosystem and its contents. To that end, LIPI has collections of flora and fauna from all over Indonesia collected and recultivated in its 5 botanical gardens: Bogor Botanical Garden, Cibodas Botanical Garden, Purwodadi Botanical Gardens, Bali Botanical Gardens, and Cibinong Botanical Garden. LIPI manages world-class flora-fauna collections. The Bogorienza Herbarium Museum established in 1817, is the third largest herbarium museum in the world. The museum has the 10th largest reference collection of fauna specimens in the world, and Indonesia Culture Collection, or INA-CC, which holds LIPI's collection of microorganisms listed in the World Microorganism Collection Culture Federation. Given its vast resources, LIPI is also appointed as a scientific authority in 1. Guiding, training, assessing, and authorizing national researchers, 2. Accrediting scientific journals, 3. Studying and evaluating biodiversity, establishing botanical gardens, as well as scientific testing and measurement. LIPI also plays a strategic role in various international scientific activities. This is evidenced by LIPI occupying the trusted position as the international focal point for UNESCO and has 3 international centers, namely Asia Pacific Centers for Ecohydrology, APCE, International Center for Interdisciplinary and Advanced Research, ICR, and Marine Biodiversity and Ecosystem Health Center, or MARBEST Center, engaged in marine training. In addition, LIPI is also active in other international organizations, such as Science Council of Asia, SCA, Botanic Gardens Conservation International, BGCI, International Council for Science, ICSU, and others. LIPI works with the following stakeholders.
Good morning. Assalamualaikum warahmatullahi wabarakatuh. Our plenary speakers, moderators, and all of distinguished participants. It's a great pleasure for me to welcome all of you on the second day of International Symposium on Applied Chemistry 2020. This day, we have a plenary session too, and also two parallel sessions at noon. But before we start this day, I want to tell all of you the several announcements to all of the participants and also our keynote speakers and moderators. First, please all of you have a stable internet connections. Make sure that computer has a working camera and audio device. In our symposium, the host will automatically mute the audio all of the participants, and the chat room is only available for delivering questions to the ongoing discussion in the session, requesting for changing or moving to the other breakout room in the parallel session, and announcement that delivered by organizing committees. Dear keynote speakers and also all of participants, don't forget to fulfill the list of attendees that we share the link in the chat rooms. And please properly rename your Zoom ID according to your participation category to set you up to the correct breakout room in the parallel sessions. For the participants that want to asking question, please state your name of institutions and also the panelist name that you want to deliver the questions. Okay, ladies and gentlemen, we will start the plenary session 2. In the plenary session, we have 3 speakers. First, Nilesh Prakash Nirmal, PhD, from Mahidol University, Thailand. Associate Professor Dr. Nurul Huda from University Malaysia Sabah, Malaysia. Professor Nina Artanti, PhD, from the Center for Chemistry, LIPI. This plenary session will moderate by Dr. Renat Andri Frediansyah, a researcher from Research Unit for Natural Product Technology, LIPI. Dr. Renat Andri Frediansyah graduated from his PhD in pharmaceutics from University of Tübingen, German, during 2007 until 2020. During 2017 until 2020. His master's degree got from Chulalongkorn Graduate Institute of Thailand during 2013 until 2015. As a young scientist, Dr. Andri got many awards. First, ASEAN Yale Summit Fellowship in National University of Singapore, and also Best Student Research Award with the Japan Society for Environmental Chemistry, and also many awards. Well, ladies and gentlemen, I will give time to our moderators, Dr. Renat Andri Frediansyah, to conduct this plenary session.
Thank you, Dr. Marisa Angelina, for your very generous introduction. Ladies and gentlemen, welcome to the second plenary session of the 6th International Conference on Applied Chemistry, focusing on natural medicine and also nutraceutical. I am Andri and I will be your moderator for this plenary session. The session has 3 keynote speakers from 3 different countries. First, Thailand, and then Malaysia, and our country, Indonesia. Before we begin, I would like to give information to all participants. At the end of 3 talks, we will have Q&A session for about 30 minutes. So if you have a question or something to be discussed, you could put your question in Zoom chat. Alternatively, you can press the raise hand button from the Zoom apps at the end of the 3 speakers. Okay, I think we're ready for the first speakers. The first keynote speaker is food chemist from Thailand. He is a Dr. Nilesh Prakash Nirmal. He is going to deliver a talk entitled Biological Activities of Fish Protein Hydrolysate. Before Dr. Nirmal starts his talk, I would like to give an overview of his background. Dr. Nirmal is foreign expert at the Institute of Nutrition from Mahidol University, Salaya, Bangkok, Thailand. He achieved his PhD in food chemistry from Prince of Songkhla University, Thailand in 2011. He was also a team leader for Wildbread in Brisbane, Australia back in 2018 to 2019. Dr. Nirmal also research fellow at the University of Queensland, Australia from 2015 up to 2018. His research interest is bioactive from plant and their derivative as alternative sources of food and food additive. He also interested in developing functional food product, food nanotechnology, seafood or agri-waste utilization, and also nanoscience. Dr. Nirmal also actively published his research in the top international journal, and currently he has a more than 35 journal. And ladies and gentlemen, please welcome Dr. Nilesh Prakash Nirmal. The time is yours and you have maximum 40 minutes to deliver your talk from now. Dr. Nirmal? Yes. Doctor, are you hear me?
Hello.
Yeah.
Hello.
The time is yours.
Audible? Am I audible?
Yes. Now, yes.
Yeah. Thank you. Thank you, Andrew, for your great introduction. And I would like to thanks the LIPI organizing committee for inviting me to this session. Can you see my presentation?
Not
Angie?
yet.
Yeah, can you see
Not
my
yet.
presentation? Yeah, okay.
Now, yes. Yeah, now, yes.
Okay.
You can start, yes.
Yeah. Well, I would like to thanks organizing committee for inviting me to give a talk on this occasion of 6th International Symposium on Applied Chemistry, focusing on natural medicine and nutraceutical. So today I'm going to talk on a biological activity of fish protein hydrolysis. So I choose this topic because we know all over the world there are a lot of people now who is moving from like animal proteins to the seafood proteins, mostly like aquaculture or fisheries production. Okay, so the fish, they are moving towards more towards fish protein because of delicacy and easily digestible protein. So what we're gonna see in this topic today is the introduction, and then sources for fish protein hydrolysis, then production, purification and identification of any peptides, and their various biological activities. Further, we will see the structure activity relationship of fish protein hydrolysis, and additionally, we will see what are the challenges for large-scale production and commercialization of this fish protein hydrolysis, and the research gap in nutraceutical applications, and finally, conclusion on this topic. So in introduction, as I told you that nowadays many people are moving towards the fish sources, seafood sources for the protein, these fish and fish products are well known because of their easily digestible protein, and the global fish production increases everywhere, including aquaculture and fisheries, like the catching fish from seas, and the aquaculture like producing fish or others seafood in aquaculture like that. So we can see in table 1 the world fisheries and aquaculture production, which is produced and presented by the FAO in during 2020. From table we can see the aquaculture and world fishery production increases steadily from 2006 until 2018. We can see all the even sea capture or aquaculture production is keep on increasing because of demand from the consumers. They demand more and more more and more fish product because of highly easily available protein and nutrients and everything compared to other protein sources. So yeah, here we can see in 2018, the total production reached until 179 million ton, among which almost 156 million ton is consumed directly for human consumption. So that means it's all directly utilized for um, human food. But the rest, like the rest of is like use around 22 million ton is used for other sources like making feed product or animal feed product or other things. So in this, almost 27 of the total fish capture is unutilized because of loss of quality or storage problem or loss of quality. So somehow this 27% from this 170 million ton fish, they are unutilized. That means they cannot sell in the market. So they have to either dump into the sea or they have to find the alternative source for that. So, and then remaining harvested fish go through the processing plant, where only 20 to 50% portion of the fish is utilized as an edible product, and the remaining parts like 80 to 50% again discarded as a byproduct or leftover. So what this leftover or byproduct contain? This leftover or byproduct contain like skin, head, viscera, bones, scales, and fins. Because obviously we just like fillets and protein part. But how about this waste? This is completely waste. So some portion is used for the animal feed production, or if someone they don't have any technology or anything, they just dump in the land or sea anywhere. So it causes like environmental hazardness. So what we can use this either unutilized fish or the fish processing waste. So these byproduct actually contain 58% of protein and 90% of fat and some content of minerals. So it has the potential to become reutilized. So we can reuse this waste, we can make some process, and yeah, we can utilize either for again for animal feed production or for human consumption again. So what we plan here, like these uh, these waste or waste product can be utilized to produce the fish protein hydrolyzate. So fish protein hydrolyzate, so far there are a lot of research that it shows the fish protein hydrolyzate containing active peptide exhibits various biological activities. So now we will see what are the sources. So as I told you, sources, it can be fish flesh or muscle, like we usually eat directly, but we can use that flesh or muscle to produce a fish protein hydrolyzate also. The second is fish skin, and then other byproduct, which include like scale, viscera, head, and fins. So fish muscle or fish flesh contain the myofibril protein or sarcoplasmic protein. Then fish skin is high in collagen. Similarly, this scale and head, they also have other proteins, including collagen and other protein sources. So we can use these protein to produce a fish protein hydrolyzate. So for this, what are the production technology? How we can produce the fish protein hydrolyzate from all these raw materials? So raw materials, it can be either whatever available for you, or you want to utilize the waste, or you want to make from flesh or anything. So there are 4 different methods so far, like people are using mostly for the production of fish protein hydrolyzate. The first, enzymatic hydrolysis. Enzymatic hydrolysis. So enzymatic hydrolysis contain, obviously as the name shows, enzymatic. That means they use enzymes for degradation of proteins. So enzymatic, so there are different kinds of enzymatic like endopeptidase, exopeptidase, and all that. And now we can get in the market like alkalase, Protamax, Nutrase, and there are a lot of varieties of proteins, commercial enzymes available, which you can use in the process of enzymatic hydrolysis. The second is chemical hydrolysis. Chemical hydrolysis, the very first, they use like acid hydrolysis or alkaline hydrolysis, but these are like really hazardous to operators and environmental also. So chemical hydrolysis, you can use either alkali or acids. And the third is auto hydrolysis. So what is auto hydrolysis? Auto hydrolysis is using the enzyme which are already present with the raw material. Every organism or everything, they have a proteolytic enzyme. They just give the active condition, including pH and temperature. So these enzymes get activated, and these proteolytic enzymes, they degraded the protein itself in the same part. And the fourth one is a fermentation hydrolysis. So fermentation, as it says, fermentation. So fermentation, they use a different bacteria to ferment the medium, and the medium contains either your fishery waste or any like whatever you want to get the hydrolyzed. So there is like again, you have to get the active pH and some mineral contents of the media so that the bacteria can grow and produce. Again, there is a proteolytic enzyme or a protease enzyme, variety of protease enzyme depend on the bacteria source you use. They produce all these enzymes, and when they produce the enzyme, these enzymes cleave the raw material, which is like very now complex, but they utilize as their growth time increase, they cleave and they produce the small fragment like peptides from the proteins. And then so this is the four way that we can produce the hydrolyzed from protein, large molecule protein to a hydrolyzed. So what they generally do is we collect the fish source, and then we homogenize raw material with the water, adjust the pH and temperature as per the requirement. If you are going to use like enzymes from external or autolysis or fermentation, so we need this pH and temperature active so that these enzymes can be active and it can completely degrade the protein. Then addition of enzymes and hydrolysis. Then we have to add the enzymes, and then there is a particular time depend on the procedure and everything. We have to allow to completely hydrolyze the protein into the hydrolyzed. Once you make sure that now the hydrolysis is done, then we can deactivate enzyme. We can deactivate enzyme by changing the temperature or by changing the pH. Once the deactivation is done, centrifuge hold the content and then collect the fractions, collect the filtrate, and that filtrate is your fish protein hydrolyzed. Actually, it's a protein hydrolyzed, but because we use the fish sources, so we call it as a fish protein hydrolyzed. So what is the next step after this? The next step contains the purification and identification. So now it is depend on your research interest that whether you want to go for purified or you just want to use like a crude, whatever you prepare fish protein hydrolyzed. But now, because due to a lot of competitions, many researchers, they wanted to purify, they wanted to identify what exactly the peptide sequence which are giving a particular biological activity. So that is the interest of scientists. So for that, they go for the purification. So purification obviously follows the chromatographic techniques, which control filtration, ultrafiltration, gel filtration, size exclusion, affinity. And there is one more I would like to add, like any enzyme bioactivity guided assay. Okay, so we can use that technique also to purify your peptide. So all these chromatographic techniques like size exclusion, which can exclude the smaller size first and retain the large molecule. So this is what affinity, like based on the positive and negative charge. Gel filtration also work on the same principles. So once you separate your peptide or the fraction of your peptides, and you then once you get those fractions of peptides, we gonna screen the activity, biological activity. Suppose you are looking for antioxidant activity. So we just check the antioxidant activity of each fractions, and wherever you get the highest antioxidant activity, we use that fraction for further identification or further high purification. So for that, we go with the identification, go with the LCMS and Maltitop. LCMS is the liquid chromatography coupled with mass spectrometry. Maltitop is the metric assisted laser deposition ionization time of flight technique. So these techniques are work on the like ionization of the molecules, and then they detect that molecule and molecular weight and everything. So here you can get actually the molecular weight, also the peptides, and the sequence also you can get. There are another techniques like peptide sequence and all, you can do that also. So the next step is the biological activity. As I told you, this fish protein hydrolyzed. So they shows the various biological activities. So far, a lot of biological activities have been screened, and there are still new biological activities going to be screened by researchers all around the world. So biological activity of the proteins are influenced by the primary structure of protein stability, amino acid composition, and content of that amino acids in the peptide sequence. Additionally, the bioactivity of protein hydrolysis depend on various conditions used during the hydrolysis. As I talk about the production, we can see that during the hydrolysis, like the types of enzyme, as I told you earlier, exopeptidase, endopeptidase, or the enzyme to the substrate ratio, pH, temperature, and time of hydrolysis. All these factors define the final product or the final product composition of your hydrolyzed, whether it can be a small peptides or it can be a large peptide, is almost depend on your time of hydrolysis and the purification technique you use and everything. So once you get this fish protein hydrolyzer, this fish protein exhibit for biological activity screening. So far, they have reported like majority of the activities, including antioxidant, antimicrobial, antihypertensive, calcium binding ability, anti-diabetic, immunomodulation, and anti-proliferation activity or anti-cancers activity. So this is what the overall, what fish protein hydrolyzed can give to our body, or how it can be a benefit, or how it can, why it can be used as a nutraceutical applications. There are a lot of like some miscellaneous activities also with this, we will see. Now, antioxidant activity. So these days, many, almost everyone now, they want some antioxidant. Wherever they are just looking on, oh, I'm looking for food which has high antioxidant, or we are screening for the phenolic compound from plant source or this source, which should have high antioxidants and microbial and other activity. So this antioxidant activity is very important actually, very, very important, because in normal living being, we actually the oxidation happen in every part of cells, or even in human body or plant also. Oxidation is the normal process of the life cycle. But sometimes the over oxidation can create a deadly or notorious reactive oxygen species, which is such as like superoxide anion radicals, hydroxide radical, hydrogen peroxide, and singlet oxygen. So once these excess free radical form, they are really unstable and very reactive. They can damage your cell protein, including nucleic acids, even lipid. They cause very serious damage to the cell and subsequently the human body. So that's why we need antioxidant. We need action antioxidant. That's why many people, they are looking for a food like keep healthy lifestyle and trying to get the food which can give you like antioxidants. So we mostly they go for like fruits which produce the most phytochemicals, which are known antioxidant, highly antioxidants. So another things I would like to tell you how these ROS or reactive oxygen species they form actually usually as a oxidation process. But the one important factor is stress, which increase all these things, which increase the number of reactive oxygen phase and inflammation, and it act like a precursor for all these things. Yeah, so now what these antioxidants do, they are the stable active chemical compounds. They are capable to intercept, combat, and prevent or reduce the reactive oxygen species. So how these antioxidant molecule they do? They do by different process. So usually there are three mechanisms like scavenging free radicals, like these free radicals, or quenching the singlet oxygen, and third is the chelating metal ion. So, so these are the three well-known mechanisms carried out by antioxidant compound. So well, now you might have surprised what is why chelating metal ion. Chelating meaning the having a complex or make this metal ion unavailable for the reaction. So why the metal ions? Because some oxidation reactions is occur through metal ion through Fenton reactions, and they increase the oxide superoxide radicals. So that increase like reactive oxygen species. So far, there are different analytical and cellular methods used to assess the antioxidant capacity of fish protein hydrolyzed. So what are the analytical methods? The analytical methods consist of like free radical scavenging activity like DPPH, superhydroxide, then hydroxyl radical scavenging activity, then metal scavenging activity, then ORAC. ORAC is the well-known analytical method. Plus there is like lipid peroxidation inhibition activity. These are the analytical activities which you can do in the flask on a chemical basis. And there are some cellular methods. So nowadays, many people, they prefer cellular antioxidant because it resembles more to human body or cellular activity. So cellular antioxidant activity consists of like antioxidant activity in cells, then lipid peroxidation inhibition in human red blood cell. Another one is inhibition of oxidative DNA damage or protein damage in the cells. So these activity you can conduct through cellular methods. So in that, the antioxidant mechanism of fish protein hydrolyzed largely depend on the amino acid sequence of the peptide. Now, so what is the, excuse me, the antioxidant mechanism of whatever you have hydrolyzed or purified compound is largely depend on the amino acid sequence. Yeah, obviously everyone knows protein is the long chain or amino sequence of amino acid. Protein consists of various amino acids and then it is formed as a protein. So these amino acid also has the side chain active molecules which are involved in various activity. So the most reactive amino acids are like acidic side chain, aspartate and glutamate, and sulfur containing side chains including cysteine and methionine, and aromatic side chains. Additionally, shorter peptide Shorter, that means low molecular weight peptide generally show higher antioxidant capacity compared to larger peptide or polypeptide. This is obvious because small molecular weight peptide are easy for access to the reactive oxygen molecules, or it can easily penetrate within the cells and it can easily react. However, the large polypeptide, because of large change, it has some adverse reaction here and there, and the active molecule is not available to counteract or interact with the radical ions. And the aromatic side chains, they are well known metal chelating because of phenolic ring. So they have ability to chelate with the metal ions. Now, the next one is the antimicrobial activity. So why the antimicrobial activity also? Yesterday, we have seen Professor Chaudhary. He have mentioned about the multi-drug resistance bugs. So we know how these bugs, they are deadly bugs. So researchers all around the fields or everyone, they are trying to find a natural source, including plant, herbs, and everything, getting phenolic compound to try to find some bioactive compound which can kill these microorganisms, which are really deadly to human beings. So there are other sources like this, which we call like antimicrobial peptide. So this fish protein hydrolyzed has a tendency to work against this bad microorganism, and it has the ability to kill those microorganisms which are hazardous to human being. So those bioactive peptide with negative charge and phospholipid membrane. So mostly these small protein hydrolyzate or small peptides, they are able to interact with this negative charge, and they are able to, after interaction, they can disrupt the cell wall and then cell lysis. This is how they kill these bad bugs. That's why this is a very important activity which can be shown by fish protein hydrolyzation. Another one is angiotensin 1 converting enzyme. So now hypertension. Hypertension is the leading cause of heart disease. So why angiotensin 1 converting enzyme? What is the role of this enzyme? So what this enzyme do, first I would like to tell you, this is a non-specific dipeptidyl carboxypeptide present in renin-angiotensin system. So what this renin-angiotensin system do? This system regulates arterial blood pressure and fluid hemostasis. So it regulates the blood flow, okay? And ACE is responsible for increasing blood pressure by converting angiotensin 1 into angiotensin 2, which is a vasoconstrictor, okay? And also a deactivator of protein vasodilatator. So like this, it choke your veins and everything, and which cause the hypertension. So therefore, to inhibit this ACE enzyme is commonly used as a first line of therapeutic treatment to control the hypertension. So this protein hydrolyzate, which can shows the ACE inhibitory activity, they are mostly, this inhibitory activity depend on amino acid composition and the molecular weight of the peptide. Okay, so the presence of hydrophobic amino acid, again here hydrophobic amino acid, arginine and valine at the C terminal. So please note that what we have found, most of the biological activities of peptides, they show the activity, and these peptides contain either hydrophobic or any particular amino acid at the C terminal, carbon terminal, which increase the ACE inhibitory potential of the peptide. there are some in vivo study as well. So it is not just in vitro, but there are some in vivo study uh, where fish protein hydrolyzate given to the spontaneously hypertensive rat. The content was like 600 milligram per kg body weight, which shows significant lower systolic and diastolic blood pressure. So it is not only the flask analysis or analytical process that it can inhibit the ACE inhibitor activity, but there are some mice study or rat study which shows the successfully they can lower the hypertension in hypertensive rat. Now, another activity is calcium binding activity. Now why this calcium binding activity here? Okay, as we know, calcium is an essential element for all living organisms. Calcium play a vital role in many cellular mechanisms. Even they are helpful for bone formation and then everything. So this is really important for well function of the body. So calcium deficiency is the worldwide problem owing to insufficient intake or decreasing the solubility. So actually, this is the major problem. Usually after age of 30, our calcium is, our body is not able to absorb the calcium from whatever the food we have eaten or whatever the tablet we eat. Our body is not absorb that completely calcium. So there is a problem for the solubility of calcium or bioavailability of calcium in the body. So that's how this fish protein hydrolyzate play a role here. Calcium ion bind with amino nitrogen and carbonyl oxygen atom of fish protein hydrolyzate. So once this calcium, like the major amino acid responsible for strongest calcium binding properties of peptides are aspartate and glutamate residues. So when we have this complex, this fish protein hydrolyzate and calcium complex, it has the great solubility and it's increased the bioavailability for our body. It not just like go in the waste or we cannot utilize. It increase the bioavailability to our body because it is chelated with this fish protein hydrolyzase. So this complex, fish protein hydrolyzase and calcium complex, could be an effective and cheap source as a calcium supplement. The next one is dipeptidyl 4 inhibitory activities. Now, what is the dipeptidyl inhibitory activity? Again, now this enzyme involved in diabetes, type 2 diabetes mellitus. Again, yesterday, Professor Chaudhary have talked about the diabetes also. And he have explained many compounds and how this, why it is important to find the bioactive molecule from natural source which can control all this disease. So this is now another like from natural protein hydrolyzate from fish waste or any fish sources. So this DPP4 inactivate or hypersin. These amino acids are mostly found in collagen and gelatin. So then fish protein hydrolyzate, it may protect the total circulating incretins, activate GLP secretion, decrease plasma glucose, and also improve glucose tolerance by inhibiting DPP4 activity. So most of the researchers, they not only just study analytical methods, but they did some cellular or animal study also using mice, or some studies include human beings for clinical trial. The next one is immunomodulation activity. Immunomodulation. We know the immune system. Immune system is the most important for well-being, right? Immune system is influenced by many internal and external factors, such as stress, unhealthy lifestyle, pathogens, and foreign antigens. So, modulating immune action is must for health and immunity of the body. So how to modulate this? So actually there are a lot of ways, like exercise, eating habit, many other things which can boost the immune activity. The immune response triggered by bioactive peptides considered to be one of the form of immunoregulation. So here it is, like bioactive peptide, how they boost this immunoregulation. So what these peptides do, they upregulate the immune function by different route. Upregulation of immune activity, such as increase of TGF. TGF is the transforming growth factor beta. They increase the transforming growth factor beta, and interleukin 10, also splenic NK, natural killer cell toxicity, and macrophage phagocytosis, and level of serum immunoglobulin A. So they increase all these activities of all these cells and the activity of molecules, so that immune system get boosted. By doing this, side by side, they also down-regulate the pro-inflammatory cytokines, including TNF alpha, that means tumor necrosis factor alpha, then interleukin 6 and 1 beta, then nitric oxide, PGE2, prostaglandin E2 production by deactivating NO synthase and COX mechanism. So, so far we saw all these fish protein hydrolysate or peptide, they are having activity. In each activity, their mechanism of action is different. And he have shown like how his synthetic probe can help to recover the cancer patient, and how it can reduce the anti-cancer activity and all that thing. So the same, like as I already told you, there are different sources which can work differently in every reaction. So what this cancer is defined as abnormal growth and proliferation of cell in the body. Therefore, controlling the initiation or promotion of cell proliferation is an important task for treating any malignant tumor. So this is the major step, like how to control. We have to control this malignation or initiation or promotion of cell at the start itself. So here, these peptides, they work very well. They shows this anti-proliferative activity through cell toxicity, cell cytotoxicity, and other mechanisms like apoptosis and blockage of nutrient. So you can see this is a natural, produced from a natural source, and a very like body friendly, you know. It may not have any more inflammation or something to your body, because it is prepared from natural source, and it can work through apoptosis and blockage of nutrients. So yes, it is a promising future research for this fish protein hydrolysate. Now, so these are the mostly known activity. Now, other reported activities. So there are other reports, people, as I told you before, researchers, scientists, they are still looking as these peptides, they has a versatile biological activity. What else they can do? This is the eagerness of scientists. What else they can do? So they try for different. So far, antithrombic activity, anticoagulant, then anti-obesity effect, anti-fatigue, anti-atherosclerosis, anti-aging, anti-glycation, cardioprotective, neuroprotective, hypocholesterinemic, and wound healing activity. Yes, all these activities, this fish protein hydrolysate has a capacity or has a tendency to act in all these disease condition, or to improve the health of human being. Now, so what importance, how all these fish protein hydrolysate shows this biological activity, The important is structure activity relationship of fish protein hydrolyzer. As I told you that the most important thing, the structural characteristic of fish protein hydrolysis, which includes molecular size, chain length, amino acid composition, and sequence, and their functional group. These are the most important thing, because these things, like mostly the amino acids and their functional group, they are involved in the all these biological activities. And obviously the size, because smaller size, it facilitate more interactions. It increase the interaction with particular area or disease or uh, reactive species. Such as, like, this structural activity of fish protein hydrolysis is different for different biological activity. The most bioactive peptide consists of hydrophobic amino acid in peptide sequence, basically at C terminal, carboxy terminal. So this is what they have concluded, that the most active, it means it can show various biological activity. This protein hydrolyzate has hydrophobic amino acid at the C terminal. Nevertheless, molecular size, chain length, and functional side chain of amino acid also influence the structural activity for the particular bioactivity. Now, after we seen that this fish protein hydrolysis is so much promising, it's like it's a really good medicine for most of the disease. It has ability to cover, and it has ability to improve um, many things. So what is the problem then? Why there is not many products in the market? Actually, we have some product. There are some product, but those products are like fish protein collagen hydrolyzate, but in a particular range. It is not any single peptide or anything. So what are the challenges? Challenges are the continuous availability of sufficient raw material. As we know, yes, this is the very important. To produce anything, we need the raw material, continuous raw material. Then only your any production system can be successful. The second thing is difficulty in the production of specific quality of peptide. Like I said, I want this particular peptide sequence. So it is quite difficult to produce in a large scale. So far, whatever the laboratory scale production is successful, but real practice or pilot scale study is quite different from the lateralization. Next, nutraceutical application. Still, as I told you, there are very few products, that too from Japan. They are selling some gelatin hydrolyzate or protein hydrolyzate, but they have something like only like 2000 kd of peptides or something like that. It is not any isolated peptide or something. So what is the nutraceutical research gap is the toxicological study of particular peptide sequence. Then pharmacokinetics, the one we have to know before any clinical application or nutraceutical application. We have to study all this toxicological study, pharmacokinetic study, and then long-term effect of consumption. We have to study that also. So if we consume every day, like long, like 6 months, 1 year, what will be the effect on our body? Because we will taking this as a supplement or nutraceutical adding. So what is the effect? Then large number of clinical trials for particular disease condition. The next, safety, environmental, and legal consideration of raw material and final product. Yeah, legal consideration is very important. That's how the regulatory harmonization all around the world is needed to become acceptable nutraceutical product all around the world. The conclusion. So this is the wonderful area and growing area. due to its various biological activities. Researchers are still interested. They are still utilizing new fish sources, various different parts using different processes. They're trying to get the different easy, fast, cheap processes to get the higher production and many things. There are a lot of scope for this. And then sources like unutilized fish, like there are some small fish sometimes when fishers they go for the catch of fish, they get the fish which is people like we don't eat, so they just throw it or dump it, but they can use these unutilized fish, then they can get the fish muscle, fish skin and other byproduct. This protein hydrolyzers show different in vitro and in vivo biological activity. The gap between commercial production and nutraceutical application of fish protein hydrolyzers should be overcome. The biological activity of fish protein hydrolyzers open the door for nutraceutical industry. These are the references, actually just small references I'm showing here. There are a lot of references, and moreover, you can get from our publications also. There are a lot of, you can just search my name. I work on shrimp processing waste also. So a lot of different and various application and waste utilization, which is published in Trends in Food Science and Technology this year. Yeah. So thank you very much everyone for your attention and listening me. However,
Okay.
yeah,
Yeah.
Andre, I would like to
Yes.
request you, if I have another 5 minutes or 10 minutes, I would like to promote my startup, my business here. Can you
Yeah,
allow me
we
to do
still
that?
have yeah, we still have around 6 minutes for you. Maybe you can
Okay,
start
6 minutes.
from now.
Wonderful.
Yeah.
Yeah. Thank you for allowing me to show my business. So I'm happy to tell everyone and announce that I have started the business with Hillberry Life Sciences. This Hillberry Life Science is the production, everything is in India. And what we are doing, we are a manufacturer of herbal extract, essential oil, and dietary supplement, capsule formulation. So all the materials are Indian origin, and we have the state of art of manufacturing facility with GMP, halal, FDA certification, then ISO, HACCP, GMP certification and everything. So as per your need, we can make all the supplement, including immune booster, then for bone osteoporosis and a lot. So what our services offer, we offer the best manufacturing services. We offer a contract research and development services and contract manufacturing organization, a capsule manufacturing processes, which include product design, formulation, packaging solution, stability studies, regulatory support, which is important also, depend on the country. And then investigation of source and availability of all raw materials. Not only that, we provide independent and collaborative technology development to provide tailor-made solution for customer-specific demand. So we do have our products in the market, like you can see, like Blackmore and all those, the nature products. They have their own product for each particular disease. So we also have that, and not only that, we can produce capsules or do the stability study or regulatory study for any product which you would like. So if you are interested for any quotes and order, please contact me on this email, nirmalnp21@gmail.com. And here you can see my mobile number. This is the Thailand code 66 and my mobile number 8345 72612. So here I would like to offer one more chance for those who are interested to do a business. Those who is interested to do business with us, I'm welcoming them. Please contact me. The business, like you have to get the industry contact from your country or all around the world, and let's start the business, commission-based business. So you can, this is a really good opportunity. If anyone interested, please contact me. Here is my email address and my mobile number. If you not get, then you can call, contact the Abdi and you can get my contact from him. Thank you very much. Thank you very much. And thank you, Andre, and thank you, organizing committee, for having me here. Thank you.
Thank you. Thank you, Dr. Nirmal, for your very great talk. Your talk about biological activity of fish protein hydrosolate for human health. That's very useful for everyone. And also good luck for your startup. I hope everyone could join with you.
Yes, yes.
If you have any question later, if you have any question for the audience, you can ask in the chat and we will discuss in the end. And
sure.
Because the bad connection in Malaysia, so we wait for Professor Huda until he could join with us in Zoom meeting. Therefore, we're going for the next keynote speaker from Indonesia.
Okay.
The speaker of this plenary session is from Indonesia. Yeah, I told you. Are you ready, from Professor Nina Artanti? Are you there? Just to make sure first.
Yep. Okay.
Yes. Okay.
You can hear me?
Yes. Okay. She is a Professor Dr. Nina Artanti, and she will deliver her talk about Indonesian biodiversity of source of functional food. Before Professor Artanti starts her speech, I would like to introduce her professional background. Presently, she is a research professor at Research Center for Chemistry, the Indonesian Institute of Sciences, or LIPI, Indonesia. She achieved her doctoral degree from Ehime University, Japan, in Applied Bioscience and Biotechnology. In 2007, she was also fellow holder for a short course conducted by JICA Bioindustry Group Training in Japan. She has also received a 2-month research fellow from participating in training on antioxidant bioassay and hepatoprotective at Zhejiang University, Guangzhou, China, with Professor Pan Yuangjiang and also Professor Lu Jia as a part of LIPI Zhejiang University in traditional medicine collaboration. Currently, her research interest is broadening from the biological activity of natural products and also functional food. Professor Artanti actively published her findings as a part of dissemination in more than 20 papers in Scopus, and she was also appointed as a reviewer in various top journals. And ladies and gentlemen, please welcome Professor Dr. Nina Artanti. The time is yours, and you have about 40 minutes to deliver your talk.
Thank you very much. Oh, yeah, that's right. It's okay?
Yeah.
Oh,
yes.
Still, okay. All right. I will deliver my talk about Indonesian biodiversity as the source of functional food. Indonesia is a large country. The land is 1.9 million kilometers square, and we have 17,000 islands when only 6,000 is inhabited. And for the sea territorial is also large, so then the population is about 274 million people. As this large country, we also known as one of the world mega biodiversity country. The diversity comprised of 1,500 species of algae, 80,000 fungal species, 595 species of this biodiversity will be used to find new drugs and everything. But of course, it's also a good source for functional food. Is it okay or because?
Sorry for your
Okay.
Yeah, true.
I know that is a close connection. Okay. And so what is I also think that this biodiversity should be also explored and used for the development of functional food. And what is the functional food? There are many definitions about functional food, but they all correlate to each other. That is a food that will be more than just us to have the nutrition, but also to have effect effect on health and should be consumed as a food. So it's probably differ from the nutraceutical of something that then can consume as a tablet or something. It should be good and have a taste nice and known that what in the history that start uh, calling the functional food. Unfortunately, in Indonesia, the term of functional food is not in the Badan POM, the Indonesian Drug and Administration, uh, Drug and Food Administration Association. What do they call it? Sorry, and because they call it before it's in 2013, it's the term of functional food is there, but then they move it, they simply don't want to have it anymore and just call for food with special health claim, and it's so only the one that already have a good probably clinical studies things that can be used that claim for that functional food. So there is about 15 ingredients that in the world approved as functional food compound, which is vitamin, mineral, sugar, alcohol, unsaturated fatty acid, peptide, and protein, amino acid, dietary fiber, prebiotic and probiotic, and so on. And it's also have to be a permitted health claim uh, because it's already a lot of clinical studies and clinical studies. So it's like calcium and will relate to osteoporosis, dietary fat to cancer, dietary saturated fat, and cholesterol related to coronary heart disease, and fiber containing grain products, fruit, vegetables, and good for probably reduce the cancer risk, and fruit and vegetable grain products that contain fiber, particularly the will be reduce the risk of coronary heart disease and sodium and hypertension, fruits and vegetable cancer, uh, of the digestive system. So this is the very common food and ingredients that we could found in many of our biodiversity, and we should be used to explore it. So this
Professor,
also
sorry for interruption again.
yeah. What the
Could
sound
you
is
increase again your voice? Yes, because I got chat.
sorry. Okay, there's another
Okay,
is a
thank
folate
you.
related to ISAC, sugar, alcohol. Do not increase dental caries, soy protein to risk for coronary heart disease, and phytosterol and phytostanol in the coronary heart disease addition whey protein, and also the green tea extract related to reduce coronary heart disease. And in Indonesia, there is many tuber crops that's because we probably only known about cassava and then uh, uh, potato and also like sweet potato, something like that. But actually, Indonesia is very rich in the tuber crops. There is many kind of tuber crops. So for several type of tubers in Indonesia, not utilized very well, like arrowroot, gembili, gembolo, suwak, porang, iles-iles, uwi, taro, pers, and kana. Because in major agriculture only usually just use cassava and sweet potato and also potato now, but the other is just less used. So it's actually a good source of uh, starch like inulin and also anthocyanin, glucomannan, which has low glycemic index, which is good for diabetic people. So we should use we explore the other tubers in Indonesia to use it as a source of functional food. I think our colleagues in Gunung Kidul and also in Subang also try to make the functional food based on this underutilized sugars. Maybe if I can go back, sorry, this is the ones that suwak is the same family of the bunga bangkai, and this is I think the black potatoes, and I also there's some is talas this one, and in biotech before we already tried to make the propagation with the tissue culture, and this the cassava, but we increase the beta carotene to get the more beta carotene to make it yellow, so it has a higher beta carotene content. I forgot this one. What's this? Okay. Uh, so this is also some underutilized vegetables. This is kucai, and there's this pegagan, in this kelor. I think the this labu, I don't know the English name, is more still not to utilize, and this time something like grass that's rarely used. But this is some medicinal traditional of some Indonesian underutilized vegetable that's have the medicinal or traditional use. So it can be developed into making the functional food, like for the allium, it's for antihypertensive, and also the centella that we know that have many activity. That's I think is also known as part of cosmetics, used for cosmetic, besides of the good in also for memory enhancements. And daun moringa is calor is also good for the content of protein, and besides it's good for antihypertensive, diabetes, and also heart disease. This underutilized vegetable will rich source of polyphenol and ascorbic acid and carotenoid. And we in Indonesia we eat a lot of rice, and most of us I think usually just eat the white rice. But then actually there is many variety of rice in Indonesia, and like the black rice, and some is a red rice. And now because the people more aware about the health, so a lot of people now start eating the red rice. So there is some variety of black rice here that it will be published that may be a good source of because it have a good low glycemic index compared to the white rice that's supposed to be good for diabetic people. And this is have a functional bioactivity studies conducted in antioxidant, anti-hyperlipidemics, anti-hyperglycemic, and cancer fixation. And for the functional food development, it says as a whole grain, rice bran, and cookies prototype. Okay. And we also have a lot of banana biodiversity. I think the last from 2018 or 2019 when we got the project uh, on Insinas priority research that we got the functional flagship is one of the uh, commodity that uh, we should use to develop the functional food. As the largest, Indonesia is the largest center of banana diversity in the world. More than 200 local cultivar and at least 15 wild variety of Musa acuminata. Banana belongs to the family of Musaceae that's consisting of 70 species. Then genomically, banana is divided into three genomes. Genome AA, BB, and SS. Banana fruit is useful for is alternative or staple food raw material of processed food with high carbohydrate and potassium that have potential for non-communicable disease. Okay. And one of Indonesian biodiversity that also high in carotenoids is the Pandanus conoideus. It's an endemic plant of Papua, Indonesia, and reported to be very rich in carotenoids. Carotenoid content of 9 clones of PO were analyzed, which range from 5.4 to 138.5 nanogram per milligram for alpha cryptoxanthin and 3.9 to 29.4 nanogram per milligram for beta cryptoxanthin and 3.5 to 80 for alpha carotene, 10.8 to 118 for beta carotene. These results show that for carotenoid content was very small as compared to total carotenoid content. This suggests that those four carotenoids were not a major component of Pandanus conoideus carotenoid. Using the principal component analysis, 9 clones of Pandanus conoideus can be grouped based on the proximity of its carotenoid content into group A, group B, and group C. This is where they are found. This is the called the buah merah, and it's also in Papua is traditionally used for the oil of that, so and also for food. So it is very good for try to develop as a source of functional food using this plant. And we found also that Indonesian rich on exotic fruit, and there is a publication that this they want to use the exotic fruit seeds to use it as functional food because they are rich in lipid and also fiber, protein, and not as carbohydrate. Probably useful to develop into the functional food. They contain many nutraceuticals like polyphenols and also flavonoids and tannins and phenolic acid such as quercetin and as caffeic acid. And besides, Indonesia also rich in animals with potential bioactivities. Pangolin is often smuggled to countries like China. Apparently, the pangolin is the potential to be raw material with omega-3 content in EPA and DHA that reduce cancer rates, reduce inflammation, hypertension, arthritis, and maintain brain function. But I think because of the COVID-19 pandemic, or people will be don't want to use the exotic animals like this to be eating because afraid of the possibility that they get dented, even though we are not sure about it. That the scales of Mandarin also contain tramadol, HCl, an analgesic active substance for the pain relief, as well as belief can cure fever, skin disease, and venereal disease. Eating meat we believe to be efficacious of aphrodisiac. Extract of roxin contain of alkaloids that can be used instead of morphine for pain relief. In addition, frog skin extraction also contains a substance that is able to activate the pancreas gland that can be used as an anti-diabetic drug to stimulate insulin. There are at least 347 species snakes in Indonesia. Traditionally, people use the extract of whaling snake at Bungarus candidus to lower blood pressure. Like the diversity of flowering plants, Indonesia also has a wealth of bees. Traditionally, bee venom may be used for rheumatic therapy, arthritis, lumbago, and skin disease. The bee venom contains apamine, melittin, phospholipase, hyaluronidase that suppress the nervous system, stimulate the liver, and adrenaline glands. And for the marine, from the marine, we have a lot of sea cucumber species that have a lot of bioactive compound that is long been used as food or traditionally in Asian countries. And the price is actually expensive. And actually, there is a 350 sea cucumber species from Indonesian water have been recorded, and at least 26 with economic value. And like Stichopus hermani, Talentota ananas, Talentota anax, and Holoturia fugolia, and Actinopiga mauritiana as most highly valued species. These organisms are potentially source of high value added compounds with therapeutic properties such as triterpene glycoside, carotenoids, bioactive peptides, vitamins, minerals, fatty acids, collagen, gelatin, chondroitin sulfate, and amino acids. In the recent years, health benefit effects of sea cucumber have been validated through scientific research and have shown medical values such as wound healing, neuroprotective, anti-tumor, anticoagulant, antimicrobial, and antioxidant. Its functional material lead to potential development of various food and biomedicine industries. And Indonesia also known with a lot of fermented food. And we know there's a lot of fermented food is good for the functional food because they contain of lot certain acids that's uh, good for our body. And this is some of the Indonesian fermented food that and the micro organisms that make the fermented food. Because like tempe is the Rhizopus oligoporus and the Rhizopus aureae from Paris bean, but the most common one is soybean. Although it also can make from other beans. And the ketchup also size. The sweet ketchup I think is traditionally of Indonesian because the other Asian countries mostly like the only the salty soy sauce. And also tauco is also based from kedelai, and for the sweet soy sauce is based on kedelai, but soybean, I mean, and but for the uh, salty soy sauce is usually sometimes use the fish. And oncom is also from soybean, but some is from peanut. And tapai is from singkong or cassava, and rice with fermented with Saccharomyces cerevisiae or yeast usually. But also the because it's not a pure culture, it's sometimes mixed with amylose, salty, and Rhizopus chinensis. And daddy is a fermented milk that's have lactobacillus, so the lactic acid bacteria, and also brem. It's also from using fermented using yeast, and tuak. It's also from nira, coconut, and aren. It's from fermented using the Saccharomyces cerevisiae. And nata is now the common that is from the coconut water, and it's using Acinetobacter. The red oncom is fermented of solid water of tofu by Nerosporas torpilla, and isolated from oncom has been studied for carotenoid production as an alternative for food and beverage colorant. Black oncom, produced by Rhizopus oligoporus, with raw material of peanut and peanut soybean cake mixed with ongok or tapioca flour waste. Active compounds are isoflavone, genistein, datein. And angkak is fermentation product of rice with monascus, used for natural food coloring, species, and medicine by Indonesian people who believe in pharmacological property. Monoclinic has been used in clinical therapy to lower blood cholesterol concentration. But as a functional food, it can be used as the part of the food. But if we only use it for the active compound, maybe it can be powder and then like a nutraceutical put in the capsule or something like that. And from the microorganism and in Indonesia, we found the nonomuria. It is converted inulin into the DFA3. It's with the enzyme inulin sucrotransferase. This from the 100 isolates of actinomycetes from soil sample collected from the province of East Kalimantan and West Nusa Tenggara screening for inulin fructotransferase. The supernatant of strain ID 06A0189 is found to be converted inulin into a compound having an RF similar to the reference standard of fructofuranose, with the anhydride, we call it the DFA3. This strain is identified as Nonomuria sp. It belongs to the Streptosporagiaceae family. This is the first report of inulin fructotransferase produced by this family. The DFA3 has many characteristics that are beneficial for functional food processing. It's a non-reducing sugar that is soluble in water, has half of sweetness of sucrose, is stable in acidic condition at high temperature, is resistant against the browning reaction and the non-caloric sweetener. The FH3 has several health benefits, enhancing the uptake of calcium, zinc, and magnesium in the small and large intestine of rats and humans, inhibits the hydroxy reaction of bile acid to provide secondary form, which is a risk factor for colon cancer. Therefore, the FA3 is expected to be a new prebiotic candidate. So now I will talk about the that's research in the of functional food in Research Center for Chemistry. This is a natural folic acid. It's work from Miss Agustin that I think some of you already heard her presentation yesterday, and there's another one today related to these studies. So we make the natural folic acid based on the like tempeh from soybean and also from uh, green bean and from the nixtamal of corn, and as well as some fermented vegetable using lactic acid bacteria, and then we make it as a source of natural folic acid by identifying using the LCMS to make sure that we got the certain amount of folic acid and use as a food to make cookies for babies, but we just try to make it, and maybe we should collaborate with our colleague in Subang and Gunung Kidul that's already have experience in collaboration with the Puskesmas and also to make the possible for the clinical studies. So this can be told or collaborate with small, medium price enterprises or with the industry, food industry that will be interesting to use this product. It's already make a publication, and we also make patent from that, and hopefully we can have collaboration with industry, and before probably have to testing it to make sure that it is very good for baby. And we also have studies on tempe extract. When at that time, Dr. Anastasia Fitria Devi get the grant from TWAS, and we study the Indonesian tempe extract and then see what's that antioxidant, and we also at that time actually tested for anti-cancer and anti-diabetic, and this is some the result that showing here that oil from tempeh with various fermentation times extracted with hexane without without and with pre-washing with acetone and or ethanol, and it gives the result is quite different. So that means that if we want to make this tempeh oil to use for the functional food, we probably can which one that's very good for have a high antioxidant then can be added to functional food ingredients. And we also conducted Miss Yati Mariati conducted fermented beverage from using kombucha with broccoli and then spinach. And we after the fermentation, we also use the membrane to separate it from the seed and then the repentate and ferment. And we found out that the antioxidant activity actually in the broccoli show good in permeate. That means it's a small molecule that come out from the from the membrane. That's whereas the other is a repentant. Is more the different activity. And we are analyzing this using LCMS, and probably hopefully we can also use the LCMS later to find out what actually when we use the membrane that come out from, and because they also have different activity. And we also involved in the herbal tea studies using the takar ayam or selaginella, and we try several uh, mistletoe species. That's because it's continuing my research when we are in trying to isolate what the mistletoe is usually used for traditional medicine in for cancer therapy, alternative medicine maybe. And we found we tested from for antioxidant and anti-cancer, and we found when we mix the cakar ayam and mistletoe and make tea, herbal tea preparation, the activity is better in the mixture. So it is a good, and it's because it's already like people used to, but drink this one. So, but for commercialization, maybe we need to do like the proper toxicity and activity testing. Also for the just for to make a tea, there is already some uh, uh, available in the market. So maybe we, if someone interest in to develop this one, it will be good as well. And because of the COVID-19, there is an interest of this functional food because like from this study, they show that viral docking entry is reduced by flavonoid or vitamin D3 or either iodide and organic sulfur. And also viral replication is reduced by polyphenol and vitamin C, as well as zinc and its ionophores. And innate immune defense also increased by thymus extract, probiotics, flavonoids, and sulfated polysaccharides. But for sulfated polysaccharide, I haven't make the presentation, but uh, the this year we are continuing from last year project the Insinas. Dr. Sofa also make the uh, biscuit for cookies from algae, the marine algae caulerpa, and now we're going to mix in with the microalgae is from to because we found it's have a immunomodulator activity, and one of the is a sulfated polysaccharide that they found isolate from the caulerpa. So it's also like we found that the oxidative stress can be reduced by vitamin C and vitamin D. It can be from the fruit and also fermented fruit, and also the environmental reaction of some botanical and probiotics. And hemostatic hormone system also increased by vitamin C, flavonoid, and polyphenol. So for the because this COVID-19, so
Professor,
yeah,
Professor, you have
yeah,
still 5 minutes left. Okay.
yeah. Maybe I finished before that is okay. We got the Miss Yati Mariati involved in making the guava fruit fermented with kombucha that help to increase our immune because it will be produce some the fermentation could be produce some acid that it's not formed if we just make the guava juice. And then for Miss Hani Mulyani and yeah, Setiani also try to make the herbs what call it the marmalade that contain, but it's not just the herbs. They also fermented it with lactic acid bacteria, and then make it to the make it a marmalade that can be used for the bread spread or to drink. So it's can be both to do both, and this hopefully it also increase the immune system for people so we can avoid the COVID-19. I think
And ladies and gentlemen, please welcome Professor Dr. Nurul Huda. The time is yours.
Okay, thank you, Dr. Adi, for nice introduction about me. First, I want to sorry to all of the organizing committee and also to the keynote speaker, Dr. Nima, and also Professor, my faculty. So faculty M come from Faculty of Food Science and Nutrition. So we said from Faculty of Food Science and Nutrition. So in our university, we also offer a degree in business, computer engineering, humanities and design, psychology, science and natural resources, and also sustainable agriculture and finance. Just some information about my current university. So you can go my detail, and then I will come to any collaboration from the participant to explore a lot of things that we can explore together. Please feel free to email me or to WhatsApp me for any potential collaboration between UMS and your institution. So it's an outline of my presentation. Okay, so I want to give some introduction first, followed by phases of wound healing, because in my presentation I will focus on the wound healing material. The wound healing material, and then followed by some explanation on the existing development of the wound healing science. And then we will enter the most important part is about the potential of the fish collagen hydrolysate as a wound healing material. And then also some sample how to extract out fish collagen hydrolysate from the fish material, and current study on the wound healing material from fish collagen hydrolysate. And we'll end with the challenges on aquaculture production, because some problem with the capture production. There are some of the marine fish population are affected, and then we can exploit the marine fish as usual. So why from the 1990 more aquaculture activity is encouraged by government to produce to supply enough amount of fish protein to the people, whether from the freshwater fish or aqua inland water, or from the marine matter or marine fish. So most of the so far the inland aquaculture production is much contribute from the freshwater fish, but I believe for the next decade maybe contribution from the marine aquaculture is more significant. But not all of the fish that we capture or produce from the aquaculture is dedicated for the food uses. Some of the fish, especially from the capture, is not suitable for the human consumption due to some reason. So why some of the food, some of the fish produced most of the fish produced dedicated for consumption, but some of the fish is normally used for non-food uses. And also we can see from this graph, the consumption is of the fish product is increased yearly, as in line with the increase of the population. It also some information that every year is around 46 million ton is the discharge in globally due to some problem due to some but mostly the problem related with the weakness of science and technology to use this kind of this raw material. So normally this kind of raw material is belong to a byproduct. So normally because normally during the processing of the fish material, probably Dr. Nirmal already mentioned about the byproduct is around 35%. Can be more, can be 50% from the whole body of the fish is belong to byproduct. We cannot use 100%. So far, we cannot use 100%, especially sample from the fillet industry. So field industry maybe can use only around 40 to 50% from the whole body of the fish. And then the rest is consists around 40 to 50% is belong to byproduct. That we need to think how to use this valuable material to produce more valuable product. And then we discard to the environment and make some negative effect to the environment, such as pollution. So far the current technology used by byproduct, as mentioned previously, is around 40 million tons. Is available the byproduct, whether byproduct from the fish processing industry or from the some suitable fish species due to some reason. So of course there are some technology available to use this kind of raw material. Normally use produce some food ingredient. Maybe we can produce from byproduct or from the unwanted fish species. We can produce some fish protein hydrolysis and use ingredient in our food system or the product. But we can also think to use this kind of the product to produce more valuable product with some bioactive compound. We can with some biological active compound, such as with some antioxidant activity, with anti-proliferative activity, with anti-anemia, antihypertensive, and antibacterial. So we have a choice whether we can use this kind of this byproduct or the low commercial fish species to produce a feed material or only to produce a food ingredient, or maybe we can choose to use this kind of tube material as a raw material to produce is quite viral in the internet. How the Brazilian doctor tried to treat the bone victim from the torrefied explosion with the fish skin. There is I think it's 3 to 4 years ago. Is quite viral in the internet about this news, and not only for the human but also applied to the animal. How they use directly the fish skin to treat the burn victim. But then with this idea come to the surface, how to produce how to study is more detailed about the potential of this fish skin. Actually, the fish skin is considered as a fish collagen because the main component of the fish skin is belong to collagen, and then to treat the burn victim from exploit or from any accident with the proper way, because it look like not nice if our body is covered with the fish skin. Then we need to think the proper technology to deliver this potential of fish skin or fish collagen as raw material for the wound healing process. This is the idea why currently me and my student working on the wound healing activity, wound healing potential from the byproduct of the tropical fish. So let me see about the phase of the wound healing, because once people experience with the wound problem is that need to conduct the 3 phases for the wound healing process. One is hemostasis. It can be hours, maybe 3 to 4 hours, followed by inflammation, and then proliferation can be a days or a week, and after that restoration. Restoration can be a month. This is a process from the wound healing restoration. Then each phase of wound healing process is influenced by a series of essential mediators. So it's quite a complex process. So let the medical doctor focus on this process. So we focus on the accelerator material. So because in this process is influenced by a series of essential mediators, platelets, synchronized inflammatory cells, cellular, extracellular matrix, protein, growth factor, inhibitor, and accelerator material, and collagen is part of the accelerator material. So we focus on the accelerator material. So besides the collagen as accelerator material, why is the collagen come with a potential for the accelerator material? Because the collagen fiber is a major component of the skin. Is a generally known, And then if we directly use the collagen, since the process is much faster, because the collagen is there. Our skin is need a collagen for the wound healing process. And then we expect the wound healing process will be much faster if we can provide sufficient amount of the collagen from the for the wound healing process. So beside from the collagen, previously some scientists also report the accelerator mediator not only exists from the collagen, but also from the fish oil. So fish oil also can play a role as accelerator mediator for the wound healing process, as mentioned by Karom in 2000. So from fish oil, we can also use the ointment for makeup purpose as a accelerator material, and also from the curcumin for the conjuncted with hyaluronic acid. So one of the potential accelerator material during the wound healing process. Got tendon also reported. Actually, got tendon actually belong to the collagen to stimulate the wound healing process by scientists. And also honey meal and aloe vera together also reported come with potential as a wound healing material. Chitin, shrimp, and also alginate. And so sea cucumber, I think I've mentioned by Nina previously, sea cucumber also the potential raw material for wound healing material. And also catfish and so chitosan. It's a list of the natural product that we can use as wound healing material besides the collagen. So this is the evidence that how the some natural component is able to act to accelerate the wound healing process if we treat with some potential bioactive component. So we can see from the report of the Parfin Pirzania use a milk ointment and silver and a bit 3%. So this one also the another evidence from the curcumin, curcumin conjuncted with hyaluronic acid. So hyaluronic acid we can extract from the heat of the chicken, from the red part of the heat of the chicken we can extract the hyaluronic acid. So you can see this one from the graph by Dr. Sharma. We can see the untreated one. The untreated one is a one closure is quite low. It's only around 25%. But if we treat with the animal with curcumin, and then the wound closure is much significant higher increase. And so if we similar if we treat with hyaluronic acid, if we combine together curcumin and hyaluronic acid, and then the wound closure is almost 100%. So the next evidence also from the fish oil. So meaning we can use different kind of the product to as a wound healing material, wound healing agent. This one from the oil from the fish egg, and then you can see from this result. So if the control group normally there is belong to the control treatment with only consist saline solution, and then compare with Kikaflora, the commercial. If we treat, if the animal treat with the fish egg oil, comparison control one, and also comparison commercial Kikaflora. So and then let we see the collagen, and let we see the collagen because I mentioned because I mentioned previously that actually the skin is consists collagen. So the similar structure with our skin. Our skin also consists collagen. So actually, according to collagen is actually from the wood is a glue and to produce. So actually, the collagen basically explore because it's a component tissue when we boil and to produce glue. And after that, some development of the collagen science and technology. And so far, the 29 distinct collagen type have been characterized. So collagen is considered a different type. So far, the 39 collagen type is characterized. Then all of the collagens, only 9 from the 29 is only what the is upon 9 is most belong to the most common collagen type. Is the 9 most collagen type is available in this raw material. This one to just inform to participant about the collagen. Meaning, if you talk about collagen, it's not only collagen. There are 29 type of collagen is available. And then we need to understand the structure, the characteristic of each collagen in detail to know more knowledge about collagen. So this one will happen if we explain with the animal or people explain with the wood accident. So actually, if the healthy collagen is come with the triple matrix, and this one is a healthy collagen. But if we explain with wound, some damage is take place. And sometimes it takes place, and sometimes it takes place in the triple helix. And then with addition of the collagen from outside, small molecule of the collagen, it will be able to faster, quicker copy of the damaged collagen. This is one of the scenario is proposed by scientists, how the potency of the fish species as a wound healing material. So we can start this collagen from the marine fish of the marine fish is byproduct. Byproduct you may consider from the skin, from the bone, or from the scale, and to produce a collagen. So when we produce the collagen, and then we need to hydrolysis the collagen. We need to go to next step to produce a collagen hydrolysate. And then when we produce a collagen hydrolysate, maybe with a different property, which depends to the process, depends to the enzyme applied, depends to the acid we use during the extraction. And then we can apply, we can test the produced collagen hydrolysate as a wound healing agent, whether in different form, maybe in the sponge form, cream form, hydrogel, or spray. So far there are less knowledge and less report on the potency of the fish collagen as a wound healing material. There are some report on jellyfish, Alaska polar, tilapia, some salmon, haddock. But all of the report show the acceleration of the wound healing rate by using this kind of the collagen hydrolysate. But you can see from the list of fish, most of the fish is not belong to tropical fish. Most of the fish is belong to temperate fish. It means consider maybe if the potential of our tropical fish as a raw material to produce the collagen hydrolysate with wound healing properties. So actually, this collagen hydrolysate from the fish collagen is a good chemo attractive for granular tissue formation. Promote according to previous report, promote cell proliferation and attachment, a good biocompatibility and biodegradability, and maintain tissue hemostasis as long as antigenicity. And a good best material for the potential dressing is according to the previous report. But I don't see any report from the tropical fish of this time being. Most of the raw materials used by the previous researchers is belong to the temperate fish. So maybe probably within the temperate fish and the tropical fish, there are some different in terms of the properties. So it's a way, it's the extraction method that we can apply. It's one of the methods that we can apply to extract the potent fish collagen hydrolysate. So by fish byproduct, whether from skin, bone, or scale, we need to conduct for pre-treatment to produce the acid soluble collagen. We need to remove the non-collagenous material. So we need to reduce the S content by using the DT. We also need to reduce the fat content by using the butyl alcohol if the fish fish skin come with high content, and then we produce the acid soluble collagen. So acid soluble collagen, we call it acid soluble collagen, and then we conduct the salting out, and then we produce the acid soluble collagen dry form of the acid collagen. But during the production of acid collagen, there are some pellets, precipitate. So the precipitate we can extract again. We can extract the precipitate by using the enzyme to produce the peptide soluble collagen. Then we freeze dry. And then they produce the soluble collagen, whether from the acid or peptide, and then studied by scientists. They're using, as mentioned to you, most of the fish species they use is belong to temperate fish. I don't see a report from the tropical fish for this time being. Salmon sala, and also tilapia. Whether tilapia is so available in Indonesia and Malaysia and so Thailand, but this one is belong to temperate tilapia. So and then come with evidence that the collagenous from the extract from salmon sala and tilapia is able to accelerate the healing rates and promote the wound healing by controlling the inflammatory reaction and increase the wound antigonist and collagen deposition. So similar report as on jellyfish. Jellyfish improve the structure closure is much faster than the control one as Alaska polar skin. Alaska polar can enhance the wound healing activities by oral administration in animal study using the rat. And also from the unicorn little jack skin and also to Nile tilapia skin. All of the report come to with the conclusion the potential of the fish collagen as a wound healing material. But unfortunately, as mentioned previously, all of the raw material fish species used is not belong to the tropical fish. It's all belong to the temperate fish. And then it's our time now, it's our high time now to explore the potential of the byproduct from the tropical marine fish or tropical freshwater fish as a wound healing agent. So also from the giant cockle, similar cuttlefish, blue shark, and also haddock, and also chum salmon. So there are the report that mentions that support the theory that the fish collagen is able to act as a wound healing material. This is a detail report from the Dr. Yung Chen about the ability of the giant collagen for the wound healing process. So you can see from this result the ability of the giant collagen for the to treat the animal with wound experience. So as you can see from this part, the wound closure is much faster if we treat with the peptide collagen, percent soluble collagen rather than the control one. Control meaning they use a saline solution to treat the animal. To similar report from the Morat Mandiri by using the collagen from the cuttlefish. Jadi from the cuttlefish skin and then extract the collagen hydrolysate peptide and then treat to the animal. So you can see this one is the group 1 is belong to consider the control one, group 2 and group 3. So as you can see how the cuttlefish collagen hydrolysate able to close the wound is more quickly rather compared to control group. Similar report from the shark fin, from the shark skin. Shark skin also consists of collagen as to promote the immersion of the wound healing. You can see similar report also reported there. A similar result shows the potential of the collagen hydrolysate to treat the animal with a wound experience. This one also the similar report using the salmon sala and also tilapia neotica skin. How this collagen extract from the salmon sala skin and also from tilapia skin is able to improve the healing rate of the animal if we treat with this kind of the collagen. Similar report from the top Felix. So a lot of evidence, we have evidence that the ability of the collagen from marine fish as a agent from the wood healing process as accelerator during the wood healing process. So this one also from the Alaska polar. Scapola is a raw material to produce a surimi. The main raw material to use a surimi production is belong to Alaska polar, and then people can use the byproduct from the Alaska polar skin as to produce the collagen hydrolysis and come with evidence that the collagen hydrolysis they produce from this Alaska polar skin is able to come with an alternative for the wound healing treatment. Similar thing from the salmon come with the evidence that the collagen from the cells kind of the salmon also can act as a good potential wound healing material. So it's a challenge now for us in Indonesia with a very huge amount of the fish species to explore our own fish as a potential raw material to produce a collagen peptide, collagen hydrolysis, and then to see whether the collagen peptide produced for our tropical fish able to come with a good property as a wound healing agent. So a lot of fish species now we can use such as a lizard fish, big eye snapper, or Nemipterus japonicus, screeci, or maybe different. It belong to godfish. It's a godfish. All of the fish is belong to underutilized fish. Then during the surimi production, they also produce about 40 to 30% of the byproduct. So we can use the byproduct which consists skin, scale, and bone of this kind of fish as a raw material to produce a collagen hydrolysis or collagen peptides, and then we can test as a wound healing agent. So this is my current PhD student. Yeah, we just started this project last year, but due to the pandemic, our result is not come yet. So we're not able to produce any publication yet related about the potency of the tropical marine fish. So we working with a different fish species is available in our tropical country, and then study the abilities, the characteristic of the peptide hydrolysis as a wound healing agent. But the study is on the way now. It's quite slow due to the pandemic, so we've not entered the lab really. So hopefully after the next year we can come with the evidence. So we still keep the raw materials that we use, but we try to use some potential tropical marine fish for our study. And then it's all my presentation about the potency of the fish collagen hydrolysis as a wound healing material. Okay, thank you to Dr. Adri.
Thank you very much, Professor Nurul Huda, for your nice talk. Very interesting topic related to collagen from fish sources. And now we are for the next session, which is question and answer. There are several questions in the chat room, and yeah, so all the speakers are ready today, and I will see the question in the chat room from the first one. Oh yeah, the question is for Dr. Niles Nirmal from Dodi Dharmajana. Are there any side effects of FPH as a medicinal ingredient? Maybe you can start to answer this question first. Are you hear me, Dr. Niles Nirmal?
Yeah. So, well, what is the question?
Again, the question is, Are there any side effects of FPH as a medicinal ingredient? From Dodi Dharma Jana.
Is there any what?
Side effects.
Side effect. Yeah, side effects of
FPH. Fish protein hydrolysate. Yeah.
See, like as I told, this is from the natural source, like whatever we are eating, like sometimes fish protein hydrolysate from fish place, and sometimes from the waste product. So it has side effect, but not really harsh, and it's again depend like whether you are applying it like from outside is not, but if we take as a supplement, it's depend on the human body. It's depend on each participant or each person, how they respond to the external protein source and everything. Am I answer the question? Hello?
Committee, can you show the Dodi Dharmajana? Maybe is it okay for the answer?
Hello,
or maybe I think yes. If he has more question, he will put in the chat.
As a medicinal ingredient.
Yeah. So we go to the next question question from Sri Priyatni. According to your information about FPH, do you have the project for large scale of FPH and what application that you have done?
Yeah, I think I have replied everyone through chat box, but I can reply here again.
Yeah, because maybe everyone want to know also for the answer of this question.
So yeah,
maybe they have the same question. Yes.
Yeah, we are now in the process for applying a big project, and yes, our past work shows that ACE and DPP inhibitory activity in our laboratory study. Yeah, these are the like applications in the sense of various biological activities, but we haven't did any clinical trials or anything yet.
Okay. Yeah. I think the most of question is for Dr. Nilesh Kumar, but I also want to ask from maybe Professor Artanti. Are you hear me? Hello, Professor Artanti.
I think she's not there. She turned off.
Yeah, maybe I will give you one question from me. Okay. What is the main challenge as a researcher in Indonesia to mine the potential of Indonesian biodiversity? The main challenge of a researcher as an Indonesian researcher.
I think the main challenge is that we have to, if we can, if we want to go to the industry, usually it's better if the industry is.
Can you increase your voice? Sorry,
sorry. As a researcher, I think not just in Indonesia, if we done research and we expect the industry to come to us, it's a bit difficult. But if we already have a talk or something, and then what they want, and we do the research for them, it will be useful, because sometimes the industry know better with like functional food that will be booming or something compared to us as a researcher. We just we try this, we try that, but we don't really know what's like expectation from the people, because that's why we are not become we don't have an industry. That's why I think that the challenge is to know what the industry research wants. So I think we better collaborate since the beginning, what they want, and we do the research. Especially now, if we get already the facility, and they because sometimes if we do research, but then we don't have the industry who will take our research, then it's just that. It's just probably just become a publication. Also, we do make a patent, but probably no one will buy our patent like that. So I think because it's probably as a researcher, especially.
Yes, these indigenous people, they are drinking this every time, and it really give you boost, you know. They are really good. So I believe like each country have um, their indigenous plants and everything. That knowledge should come to the public. So we really have to go and meet our indigenous people, or these people like native people. They have that knowledge. We have to get that knowledge from them, and we have to translate to research into the public awareness to make them interest. Yeah, this is really good topic.
Okay.
Thank you, Dr.
Thank you so much for your.
Yeah.
Okay, thank you.
Yeah. Okay, so we start also have another question in the chat room. Oh, maybe this question is for Professor Dr. Nurul from Ani Soliha. Dr. Nurul, you hear me?
Yes, Dr. Adi. Yes, I'm here.
Okay, yeah, the question is there any effect of freezing treatment on enzyme hydrolysis process?
Okay, thank you, Dr. Andy. So thank you for the good question. I think as mentioned, we are just on the way. We just start this project last year. So we received the highest research project approved by my university. Is with value is 600 juta rupiah. Yeah, the total budget of this is 600 juta rupiah, but start last year only. So and then during the pandemic, we're not able to come with more result now. We no more result now because one pandemic COVID coming, the university close and close again. But we expect for the for the if the piece is in the frozen the piece in the frozen form, we don't affect to the hydrolysis. So meaning whether we use the fresh fish of the frozen form fish, we not expect any different for the property of the fish collagen produced so far. But yet this one just based on my expectation only. We're not able to come with any scientific evidence for this answer. But my expectation, whether we use the frozen fish or fresh part of the fresh fish, will not affect to the hydrolysis. Okay, I think it's my answer, Dr. Adi.
Yes. Okay. Development of FPH.
Yeah. So the main problem is consistency and reproducibility. That is the main challenge in this production of FPH, fish protein hydrolysis. Yeah, because the problem is even the fish source. Even if we consider one species, the one species does not mean they have the same proportion of protein content or their body composition. So that make quite difficult to produce, get the consistency in the production, and get the like same reproducibility or batch to batch quality of the fish protein hydrolysis.
Okay, thank you. We still have more questions. I hope you're still okay with this question.
Yeah,
so another question is from Wita Kartika Restu to Dr. Niles. I am interested in short peptide from FSH and would like to ask regarding how much the yield that you get from the raw material of fish until the purification to be the
particular sequence, you know, that is okay. But if you go for purification of that particular active peptide, the yield is really low, very low. And about the hydrogel, I haven't worked on hydrogel yet, but yes, it is a really good idea. And yeah, that will be really good idea. Yes, so usually how to confirm that? So the first thing, like if you check the biological activity of your raw material and the fraction, you can see there is something like, obviously the raw material or the first fraction may not give you any result, but the fraction can give you. So it shows that something has happened. Something has happened to the raw material, which is the long polypeptide chain. It has degraded and you get the some fraction, something has happened. So next step, what you can confirm is, again, you can go for the chromatography and something that check the biological activity of each fraction. Furthermore, you can go for the mass spectrophotometry, which can give you the molecular weight. From there, yeah, you can confirm that, oh yes, I get the hydrolyzed or the smaller peptide compared to the parent peptide of polypeptide.
Thank you. And maybe I will also start question for Professor Nurul Huda from Carla USN Kolaka regarding utilization of fish collagen for wound healing. Are we need to evaluate the metal content in the sample, especially sample near mining sites? If the sample found to be contaminated with heavy metal, can we use this kind of sample?
Yes, I think for Mrs. Carla. Yeah. Yes, it's very important to us to make sure that the raw material they use production is belong to use to any process, not only to produce the fish collagen hydrolysate or the hydrolysate peptide, but also for normal consumption. It's very important if the area are decontaminated or nearby the mining area. It's very important to conduct the primary analysis of the heavy metal contamination. And especially according to some previous research, if the fish live around the area with heavy metal contamination,
there are different using. Are there any different using food between for functional food and for drugs such as for low?
I think when you say that's why it's different from food and drugs. Usually, for the functional food, you still won't be too much like that. But if you use it as a drug, usually you extract them, you boil them like until from 1 liter become only small amount. So I think the difference is in the dosage. Usually, if you want to use it as a drug, usually you use more compared to when you if it's already have lowering effect, even though you just drink like the herbal tea. If you try to do the DPPH directly from the herbal tea, it seems that you have IC50 a bit high, but you drink a lot. You drink like at least 100 ml of tea compared to if you drink drug that only 5 milligram or something. Maybe the 100 ml of tea is the same as the 5 milligram drug like that. But then that's why it's the different how to you take it. I think it will be same, but it's in different way. Is it answer your question or not? Because usually when you work with food, it will be like you measure the IC50. It won't be fair.
Dr. Nilesh Kumar, based on your research, which is the better method for producing FPH?
All right, so better method is enzymatic hydrolysis. We can the enzymatic hydrolysis can give you really small molecular peptides. So that is really good. However, again, it's depend on the what is your interest and something. And I also explained like fermentation hydrolysis, which can give you two benefits. Like it can produce protein hydrolyzate and it can produce enzyme also, like for commercial applications. So these are the, so obviously depend on your aim. If you really want to go for small molecular weight peptide, enzymatic hydrolysis is the best production process. Yeah.
Okay. Thank you. Still have question for you from Ahmad Sarifuddin Lipi. The question is, okay. Nice presentation, Dr. Nirmal. Thanks for sharing your knowledge. I ever heard that FPH has functionality to prevent high blood pressure. That is very great news for high blood pressure patient.
Yeah.
But in terms of application, because most of Indonesian or common people don't like fishy taste and aroma of FPH, is there any way to get rid of this flavor of FPH? Yeah.
Yes, yes. We have a good news. There is a way that we can avoid the fishy smells, and it can make like a plain without any smells, or we can add some other flavor which can improve the aroma and everything. There is a process called debittering process. We can use that as well. Yeah. So
thank you. Is there more question in the
yes.
chat or not? I will check again. Maybe I have a question for Professor Huda. Professor Huda there, are you there?
Yes, please.
Yeah, I just want to know, is there any difference between the product, a collagen product from marine fish and freshwater, about the quality or something else? Thank you.
Okay, thank you, Dr. Adi. I think each fish come with its own property. Each fish come with unique property. So we need to study species by species. We cannot generalize all the fish species is as one category. So why? Maybe some species is potential, maybe another species is not potential. So in general, each fish species come with its own property. So we need to study species by species. And also not only because due to the different species, but if we conduct the process differently, so the result will be different. If we use a different enzyme, and then the result is also different. So why we need to specifically. So as we need to work specifically to the species and also to the specific enzyme to produce a better quality of the collagen hydrolysis. I think it's my answer, Dr. Ali.
Thank you so much for your answer. How about from Dr. Nile's perspective about this? If it's from collagen, right? Collagen quality from different source like marine source and also the fresh water. How about the hydroxylate, fish protein hydroxylate? Also the same quality, or maybe you have another perspective to answer?
Yeah, as Professor Nurul already told you that yes, every species has a different composition. That yes, every species has a different compositions. So we have to study that. And obviously, as I also answered the previous questions, like the raw material is affect your final product quality. So we have to do that. So this is the basic challenge in this, Although, like Professor Nurul already introduced you.
So interested with the tuber diversity, where usually we can find the tuber diversity in Indonesia is only from the west of Indonesia or from all the part of Indonesia? Usually,
all parts of Indonesia maybe have the different kind of tubers, but the problem is like now people who eat sweet potato now also eat rice. Probably not many grown that and the wild type probably still there. So I think probably the biology people who's doing exploration know probably the biology people who's doing exploration know the centra of the, but because people don't grow it that much, because they're only black rice anymore, something like that. Because if I heard that people said, oh, we should go to organic stuff, but if you use really organic, it's not really you have enough small part of research, but not really people use it, something like that.
Okay, thank you so much for Professor Artanti. And I think I did not see more questions in the chat. So I think that's all for the second plenary session for today. Thanks for all your contribution to the discussion. Also thanks to Dr. Nirmal, Professor Huda, and also Professor Artanti for your valuable time at this conference. See you other times. And now I will give back the time to Dr. Marisa. Dr. Marisa, the time is yours. Thank you.
Yes. Yes, Dr. Andri, thank you for the nice leading in the sessions. And also we are thank you to all. Please turn on your video. The committee will take this will capture the screens. Okay, we wait for all of you to open the video. Okay, I will count 1, 2, 3. Okay, please wait. 1, 2, and 3. Okay, thank you.
Thank you.
Also, thanks to all of participants that I think when I see the list of participants is in this day, the same with the first day of our symposium. We are really grateful that you still attend our symposium till the second day of symposium. I will tell to you, all of you, about our next agenda. The next agenda, we have 2 parallel sessions. Will be start from 1 p.m. until 3 p.m. The first parallel session will be divided into 2 sessions, session 3 and session 4. The session 3 contain 3 rooms. The first room of parallel session will be discussed about natural product and traditional medicines with the moderator Andi Febrisiantosa, PhD. And also the second room will be discussed about analytical chemistry and chemical engineering and will be led by Dr. Osi Arutanti, Master of Science. The third room will be discussed about food technology and functional food, will be moderated by Anastasia Fitria Devi, PhD. And the second parallel session will be start on 3:15 p.m. until 4:45 p.m. and also will be divided into 3 rooms. The first room will be discussed about pharmaceutical chemistry, pharmaceutical technology, and drug delivery system. It will be led by Faiza Mariani, Master of Science. And the second room will be discussed about analytical chemistry and chemical engineering. Will be moderated by Dilani Putri Ramadan Tias, Master of Science. And also the third room will be discussed about analytical chemistry.
See you. Okay.
Yeah, see you.
Thank you, everyone. Bye.
Becoming a world-class research institution that uses research to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences, or LIPI. LIPI was formally established in 1967, but its history goes back as far as 1817 with the establishment of a plant conservation center at the Bogor Botanical Garden. We are spread over 11 provinces. We have over 4,800 employees. We are the largest research institute and the largest patent contributor in Indonesia. LIPI is directly responsible to the President of the Republic of Indonesia, with the coordination of the Ministry of Research, Technology, and Higher Education. LIPI's acting chairman is Professor Dr. Bambang Subianto, who is assisted by the Vice Chairman, General Secretary, and 5 deputies who each oversee research centers and technical service units in their respective fields. LIPI has a strong foundation built on conducting research that delivers large-scale, significant, and real results for Indonesia. LIPI's research focus areas include life sciences, consisting of molecular biology, biotechnology and medicine, biomedical science, molecular and post-genomic agriculture, exploration and utilization of biodiversity research, earth sciences, consisting of marine biology and inland waterways, eco-hydrology, coastal and marine areas, climate change and disaster management, and mineral resources research. Engineering sciences, consisting of defense and IT security, telecommunications and electronics, eco-friendly transportation, advanced materials and nanotechnology research. Social sciences and humanities, consisting of people and population, culture and language, policy and governance or good governance, local government and regional Since 2010, more than 171 MOUs of cooperation have been established between LIPI and the national stakeholders. This cooperation has resulted in the establishment of various science and techno parks. These parks incubate technology startups and facilitate technology transfer. LIPI has also established more than 70 partnerships with international partners since 2010 in various branches of science across the world. Including research collaboration, workshops, and exchange programs and scholarships for LIPI researchers. For more than 50 years, LIPI has produced research that has significantly improved the quality of life of Indonesians, as well as the development of science in Indonesia. To encourage the spirit of research among the younger generation, LIPI has, since 1968, hosted research paper competitions for teenagers, as well as the National Young Inventor Award, which is keenly followed by students throughout Indonesia. We are ready to cooperate with various elements, both national and international, to realize our vision of becoming a world-class research institution to improve the competitiveness of Indonesia in the future.
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and brongkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, brongkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressure steam as a sterilizing agent. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of Good Manufacturing Process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
Each vision begins with a spark of inspiration. And each dream is driven by the passion to realize it. For over 50 years, we have worked hard to serve Indonesia. Becoming a world-class research institution that uses research to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences, or LIPI. LIPI was formally established in 1967, but its history goes back as far as 1817, with the establishment of a plant conservation center at the Bogor Botanical Garden. We are spread over 11 provinces. We have over 4,800 employees. We are the largest research institute and the largest patent contributor in Indonesia. LIPI is directly responsible to the President of the Republic of Indonesia with the coordination of the Ministry of Research, Technology, and Higher Education. LIPI's acting chairman is Professor Dr. Bambang Subianto, who is assisted by the Vice Chairman, General Secretary, and 5 deputies who each oversee research centers and technical service units in their respective fields. LIPI has a strong foundation built on conducting research that delivers large-scale, significant, and real results for Indonesia. LIPI's research focus areas include life sciences, consisting of molecular biology, biotechnology and medicine, biomedical science, molecular and post-genomic agriculture, exploration and utilization of biodiversity research, earth sciences, consisting of marine biology and inland waterways, eco-hydrology, coastal and marine areas, climate change and disaster management, and mineral resources research. Engineering sciences, consisting of defense and IT security, telecommunications and electronics, eco-friendly transportation, advanced materials and nanotechnology research. Social sciences and humanities, consisting of people and population, culture and language, policy and governance, or good governance, local government and regional autonomy, social conflict, economics and poverty research, scientific services, consisting of scientific repository, metrology and standardization, innovation of technology transfer and utilization of science and technology services. Studies into scientific and technological developments and scientific community training. LIPI researchers are supported by world-class facilities spread over 48 research centers and technical implementation units throughout Indonesia. LIPI also has research vessels for oceanography research activities, as well as other marine research expeditions. We understand the importance of maintaining the natural ecosystem and its contents. To that end, LIPI has collections of flora and fauna from all over Indonesia collected and recultivated in its 5 botanical gardens: Bogor Botanical Garden, Cibodas Botanical Garden, Purwodadi Botanical Gardens, Bali Botanical Gardens, and Cibinong Botanical Garden. LIPI manages world-class flora-fauna collections. The Bogoriense Herbarium Museum, established in 1817, is the 3rd largest herbarium museum in the world. The museum has the 10th largest reference collection of fauna specimens in the world, and Indonesia Culture Collection, or INA-CC, which holds LIPI's collection of microorganisms listed in the World Microorganism Collection Culture Federation. Given its vast resources, LIPI is also appointed as a scientific authority in 1. Guiding, training, assessing, and authorizing national researchers, 2. Accrediting scientific journals, 3. Studying and evaluating biodiversity, establishing botanical gardens, as well as scientific testing and measurement. LIPI also plays a strategic role in various international scientific activities. This is evidenced by LIPI occupying the trusted position as the international focal point for UNESCO and has 3 international centers, namely Asia-Pacific Centers for Eco-Hydrology, APCE, International Center for Interdisciplinary and Advanced Research, ICR, and Marine Biodiversity and Ecosystem Health Center, or MARBEST Center, engaged in marine training. In addition, LIPI is also active in other international organizations, such as Science Council of Asia, SCA, Botanic Gardens Conservation International, BGCI, International Council for Science, ICSU, and others. LIPI works with the following stakeholders to disseminate its research results to the community, namely national ministries and state-owned enterprises in the field of metrology and testing, local government, universities, and industries, including the private sector. Since 2010, more than 171 MOUs of cooperation have been established between LIPI and the national stakeholders. This cooperation has resulted in the establishment of various science and techno parks. These parks incubate technology startups and facilitate technology transfer. LIPI has also established more than 70 partnerships with international partners since 2010 in various branches of science across the world. Including research collaboration, workshops, and exchange programs and scholarships for LIPI researchers. For more than 50 years, LIPI has produced research that has significantly improved the quality of life of Indonesians, as well as the development of science in Indonesia. To encourage the spirit of research among the younger generation, LIPI has, since 1968, hosted research paper competitions for teenagers, as well as the National Young Inventor Award, which is keenly followed by students throughout Indonesia. We are ready to cooperate with various elements, both national and international, to realize our vision of becoming a world-class research institution to improve the competitiveness of Indonesia in the future.
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and bronkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, bronkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as a sterilizing agent. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of Good Manufacturing Process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
Each vision begins with a spark of inspiration. And each dream is driven by the passion to realize it. For over 50 years, we have worked hard to serve Indonesia. Becoming a world-class research institution that uses research to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences, or LIPI, the largest patent contributor in Indonesia. LIPI is directly responsible to the President of the Republic of Indonesia, with the coordination of the Ministry of Research, Technology, and Higher Education. LIPI's acting chairman is Professor Dr. Bam.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, nama saya Agus Ketut Sunardianto, peneliti dari Pusat Penelitian Dunia Lembaga Ilmu Pengetahuan Indonesia. Saya sekarang berada di Belanda, sedang mengikuti program postdoktoral saya di Delft, Belanda. Pengalaman saya mengikuti konferensi ISAC 2019, yaitu satu, saya diberikan kesempatan untuk mempresentasikan hasil penelitian saya secara efektif. di mana dari presentasi tersebut saya mendapatkan umpan balik dan komen dari para peserta lain, di mana umpan balik tersebut sangat bermanfaat bagi saya untuk menimbulkan ide-ide baru melanjutkan penelitian saya. Kemudian manfaat yang kedua adalah dengan menghadiri konferensi ISAC 2019, saya juga diberi kesempatan untuk melihat secara langsung bagaimana cara berpresentasi dari peserta lain yang bidangnya berbeda dengan saya atau bidangnya sama dengan saya. Oleh sebab itu, dari situ saya mendapatkan pengetahuan baru terkait dengan topik-topik penelitian terbaru dan terhangat di Indonesia. Kemudian yang ketiga adalah konferensi ISAC Internasional 2019 merupakan ajang bertemunya para peneliti, kemudian para dosen dan juga akademisi serta dari kalangan industri, sehingga tercipta kolaborasi berkelanjutan untuk kebermanfaatan ilmu kepada masyarakat luas. Demikian dari saya, salam ISAC 2020. Wassalamualaikum warahmatullahi wabarakatuh.
ISAC, Indonesian Science Expo, is one of the biggest scientific festival in Indonesia. There are many scientific activities in this event, such as conferences, exhibition, and scientific competition, particularly for young people. In ISAC 2019, Econ Probios, one of the member ISAC conference, got many benefits from interacting with various scientific stakeholders in that event. However, now we got different conditions. In ISAC 2020, we cannot enjoy directly, but it held in virtually. So stay healthy in COVID-19 pandemic situation, and you still got benefit from ISAC 2020 by infinity science exploration. Thank you.
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and bronkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage the process. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as a sterilizing agent. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of Good Manufacturing Process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, nama saya Agus Ketut Sunardianto, peneliti dari Pusat Penelitian Ilmiah Lembaga Ilmu Pengetahuan Indonesia. Saya sekarang berada di Belanda, sedang mengikuti program postdoktoral saya di Delft, Belanda. Pengalaman saya mengikuti konferensi ISAC 2019, yaitu satu, saya diberikan kesempatan untuk mempresentasikan hasil penelitian saya secara efektif, di mana dari presentasi tersebut saya mendapatkan umpan balik dan komen dari para peserta lain, di mana umpan balik tersebut sangat bermanfaat bagi saya untuk menimbulkan ide-ide baru melanjutkan penelitian saya. Kemudian manfaat yang kedua adalah dengan menghadiri konferensi ISAC 2019, saya juga diberi kesempatan untuk melihat secara langsung bagaimana cara berpresentasi dari peserta lain yang bidangnya berbeda sendiri. Demikian dari saya mengucapkan selamat datang kepada semuanya di acara International Conference ISAC 2020. Semoga presentasi Bapak Ibu semuanya lancar dan acara ISAC juga lancar, sehingga nantinya selain mendapatkan ilmu dan bermanfaat, Bapak Ibu juga dapat menjalin kolaborasi berkelanjutan untuk kebermanfaatan ilmu kepada masyarakat luas. Demikian dari saya. Salam ISAC 2020. Wassalamualaikum warahmatullahi wabarakatuh.
ISAC, Indonesian Science Expo, is the one of the biggest scientific festival in Indonesia. There are many scientific activities in this event, such as conferences, exhibition, and scientific competition, particularly for young people. In ISAC 2019, Econ Probios, one of the member ISAC conference, got many benefits from interacting with various scientific stakeholders in that event. However, now we got different conditions. In ISAC 2020, we cannot enjoy directly, but it held in virtually. So, stay healthy in COVID-19 pandemic situation, and you still got benefit from ISAC 2020 by infinity science exploration. Thank you.
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and brongkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, brongkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of Good Manufacturing Process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, nama saya Agus Ketut Sunardianto, peneliti dari Pusat Penelitian Ilmiah Lembaga Ilmu Pengetahuan Indonesia. Saya sekarang berada di Belanda, sedang mengikuti program postdoktoral saya di Delft, Belanda. Pengalaman saya mengikuti konferensi ISAC 2019, yaitu 1. Saya diberikan kesempatan untuk mempresentasikan hasil penelitian saya secara efektif, di mana dari presentasi tersebut saya mendapatkan umpan balik dan komen dari para peserta lain, di mana umpan balik tersebut sangat bermanfaat bagi saya untuk menimbulkan ide-ide baru melanjutkan penelitian saya. Kemudian manfaat yang kedua adalah dengan menghadiri konferensi ISAC 2019, saya juga diberi kesempatan untuk melihat secara langsung bagaimana cara berpresentasi dari peserta lain yang bidangnya berbeda dengan saya atau bidangnya sama dengan saya. Oleh sebab itu, dari situ saya mendapatkan pengetahuan baru terkait dengan topik-topik penelitian terbaru dan terhangat di Indonesia. Kemudian yang ketiga adalah konferensi ISAC Internasional 2019 merupakan ajang bertemunya para peneliti, kemudian para dosen dan juga akademisi serta dari kalangan industri, sehingga tercipta kolaborasi yang sangat kuat yang harapannya bisa membawa dampak kepada masyarakat luas dan juga mengembangkan ilmu pengetahuan itu sendiri. Demikian dari saya mengucapkan selamat datang kepada Bapak Ibu semuanya di acara International Conference ISAC 2020. Semoga presentasi Bapak Ibu semuanya lancar dan acara ISAC juga lancar, sehingga nantinya selain mendapatkan ilmu yang bermanfaat saya. Salam ISAC 2020. Wassalamualaikum warahmatullahi wabarakatuh.
ISAC, Indonesian Science Expo, is the one of the biggest scientific festival in Indonesia. There are many scientific activities in this event, such as conferences, exhibition, and scientific competition, particularly for young people. In ISAC 2019, Econ Probios, one of the member ISAC conference, got many benefits from interacting with various scientific stakeholders in that event. However, now we got different conditions. In ISAC 2020, we cannot enjoy directly, but it held in virtually. So, stay healthy in COVID-19 pandemic situation, and you still got benefit from ISAC 2020 by infinity science exploration. Thank you.
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and bronkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, bronkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as a sterilizing agent. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of Good Manufacturing Process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, nama saya Agus Ketut Sunardianto, peneliti dari Pusat Penelitian Ilmiah Lembaga Ilmu Pengetahuan Indonesia. Saya sekarang berada di Belanda, sedang mengikuti program postdoktoral saya di Delft, Belanda. Pengalaman saya mengikuti konferensi ISAC 2019, yaitu satu, saya diberikan kesempatan untuk mempresentasikan hasil penelitian saya secara efektif, di mana dari presentasi tersebut saya mendapatkan umpan balik dan komen dari para peserta lain, di mana umpan balik tersebut sangat bermanfaat bagi saya untuk menimbulkan ide-ide baru melanjutkan penelitian saya. Kemudian manfaat yang kedua adalah dengan menghadiri konferensi ISAC 2019, saya juga diberi kesempatan untuk melihat secara langsung bagaimana cara berpresentasi dari peserta lain yang bidangnya berbeda dengan saya atau bidangnya sama dengan saya. Oleh sebab itu, dari situ saya mendapatkan pengetahuan baru terkait dengan topik-topik penelitian terbaru dan terhangat di Indonesia. Kemudian yang ketiga adalah konferensi ISAC Internasional 2019 merupakan ajang bertemunya para peneliti, kemudian para dosen dan juga akademisi serta dari kalangan industri, sehingga tercipta kolaborasi yang sangat kuat yang harapannya bisa membawa dampak kepada masyarakat luas dan juga mengembangkan ilmu pengetahuan itu sendiri. Demikian dari saya mengucapkan selamat datang kepada Bapak Ibu semuanya di acara International Conference ISAC 2020. Semoga presentasi Bapak Ibu semuanya lancar dan acara ISAC juga lancar, sehingga nantinya selain mendapatkan ilmu yang bermanfaat, Bapak Ibu juga dapat menjalin kolaborasi berkelanjutan untuk kemanfaatan ilmu kepada masyarakat luas. Demikian dari saya, salam ISAC 2020. Wassalamualaikum warahmatullahi wabarakatuh.
ISAC, Indonesian Science Expo, is the one of the biggest scientific festival in Indonesia. There are many scientific activities in this event, such as conferences, exhibition, and scientific competition, particularly for young people. In ISAC 2019, Econ Probios, one of the member ISAC conference, got many benefits from interacting with various scientific stakeholders in that event. However, now we got different conditions. In ISAC 2020, we cannot enjoy directly, but it held in virtually. So, stay healthy in COVID-19 pandemic situation, and you still got benefit from ISAC 2020 by infinity science exploration. Thank you.
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and brongkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, How can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, brongkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as a sterilizing agent. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of Good Manufacturing Process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, nama saya Gagus Ketut Sunardianto, peneliti dari Pusat Penelitian Ilmiah Lembaga Ilmu Pengetahuan Indonesia. Saya sekarang berada di Belanda, sedang mengikuti program postdoktoral saya di Delft, Belanda. Pengalaman saya mengikuti konferensi ISAC 2019, yaitu 1. Saya diberikan kesempatan untuk mempresentasikan hasil penelitian saya secara efektif, di mana dari presentasi tersebut saya mendapatkan umpan balik dan komen dari para peserta lain, di mana umpan balik tersebut sangat bermanfaat bagi saya untuk menimbulkan ide-ide baru melanjutkan penelitian saya. Kemudian manfaat yang kedua adalah dengan menghadiri konferensi ISAC 2019, saya juga diberi kesempatan untuk melihat secara langsung bagaimana cara berpresentasi dari peserta lain yang bidangnya berbeda dengan saya atau bidangnya sama dengan saya. Oleh sebab itu, dari situ saya mendapatkan pengetahuan baru terkait dengan topik-topik penelitian terbaru dan terhangat di Indonesia. Kemudian yang ketiga adalah konferensi ISAC Internasional 2019 merupakan ajang bertemunya para peneliti, kemudian para dosen dan juga akademisi serta dari kalangan industri, sehingga tercipta kolaborasi yang sangat kuat yang harapannya bisa membawa dampak kepada masyarakat luas dan juga mengembangkan ilmu pengetahuan itu sendiri. Demikian dari saya mengucapkan selamat datang kepada semuanya di acara International Conference ISAC 2020. Semoga presentasi Bapak Ibu semuanya lancar dan acara ISAC juga lancar, sehingga nantinya selain mendapatkan ilmu yang bermanfaat, Bapak Ibu juga dapat menjalin kolaborasi berkelanjutan untuk pemanfaatan ilmu kepada masyarakat luas. Demikian dari saya, salam ISAC 2020. Wassalamualaikum warahmatullahi wabarakatuh.
ISAC, Indonesian Science Expo, is the one of the biggest scientific festival in Indonesia. There are many scientific activities in this event, such as conferences, exhibition, and scientific competition, particularly for young people. In ISAC 2019, Econ Probios, one of the member ISAC conference, got many benefits from interacting with various scientific stakeholders in that event. However, now we got different conditions. In ISAC 2020, we cannot enjoy directly, but it held in virtually. So, stay healthy in COVID-19 pandemic situation, and you still got benefit from ISAC 2020 by infinity science exploration. Thank you.
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and brongkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, brongkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as a sterilizing agent. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of Good Manufacturing Process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, nama saya Agus Ketut Sunardianto, peneliti dari Pusat Penelitian Ilmiah Lembaga Ilmu Pengetahuan Indonesia. Saya sekarang berada di Belanda, sedang mengikuti program postdoktoral saya di Delft, Belanda. Pengalaman saya mengikuti konferensi ISAC 2019, yaitu 1. Saya diberikan kesempatan untuk mempresentasikan hasil penelitian saya secara efektif, di mana dari presentasi tersebut saya mendapatkan umpan balik dan komen dari para peserta lain, di mana umpan balik tersebut sangat bermanfaat bagi saya untuk menimbulkan ide-ide baru melanjutkan penelitian saya. Kemudian manfaat yang kedua adalah dengan menghadiri konferensi ISAC 2019, saya juga diberi kesempatan untuk melihat secara langsung bagaimana cara berpresentasi dari peserta lain yang bidangnya berbeda dengan saya atau bidangnya sama dengan saya. Oleh sebab itu, dari situ saya mendapatkan pengetahuan baru terkait dengan topik-topik penelitian terbaru dan terhangat di Indonesia. Kemudian yang ketiga adalah konferensi ISAC Internasional 2019 merupakan ajang bertemunya para peneliti, kemudian para dosen dan juga akademisi serta dari kalangan industri, sehingga tercipta kolaborasi yang sangat kuat yang harapannya bisa membawa dampak kepada masyarakat luas dan juga mengembangkan ilmu pengetahuan itu sendiri. Demikian dari saya mengucapkan selamat datang kepada semuanya di acara International Conference ISAC 2020. Semoga persiapan Bapak Ibu semuanya lancar dan acara ISAC juga lancar, sehingga nantinya selain mendapatkan ilmu yang bermanfaat, Bapak Ibu juga dapat menjalin kolaborasi berkelanjutan untuk kebermanfaatan ilmu kepada masyarakat luas. Demikian dari saya, salam ISAC 2020. Wassalamualaikum warahmatullahi wabarakatuh.
ISAC, Indonesian Science Expo, is the one of the biggest scientific festival in Indonesia. There are many scientific activities in this event, such as conferences, exhibition, and scientific competition, particularly for young people. In ISAC 2019, Econ Probios, one of the member ISAC conference, got many benefits from interacting with various scientific stakeholders in that event. However, now we got different conditions. In ISAC 2020, we cannot enjoy directly, but it held in virtually. So, stay healthy in COVID-19 pandemic situation and you still got benefit from ISAC 2010-20 by infinity science exploration. Thank you.
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and bronkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, bronkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as a sterilizing agent. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of Good Manufacturing Process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, nama saya Gagus Ketut Sunardianto, peneliti dari Pusat Penelitian Kimia Lembaga Ilmu Pengetahuan Indonesia. Saya sekarang berada di Belanda, sedang mengikuti program postdoktoral saya di Delft, Belanda. Pengalaman saya mengikuti konferensi ISAC 2019, yaitu satu, saya diberikan kesempatan untuk mempresentasikan hasil penelitian saya secara efektif, di mana dari presentasi tersebut saya mendapatkan umpan balik dan komen dari para peserta lain, di mana umpan balik tersebut sangat bermanfaat bagi saya untuk menimbulkan ide-ide baru melanjutkan penelitian saya. Kemudian manfaat yang kedua adalah dengan menghadiri konferensi ISAC 2019, saya juga diberi kesempatan untuk melihat secara langsung bagaimana cara berpresentasi dari peserta lain yang bidangnya berbeda dengan saya atau bidangnya sama dengan saya. Oleh sebab itu, dari situ saya mendapatkan pengetahuan baru terkait dengan topik-topik penelitian terbaru dan terhangat di Indonesia. Kemudian yang ketiga adalah konferensi ISAC Internasional 2019 merupakan ajang bertemunya para peneliti, kemudian para dosen dan juga akademisi serta dari kalangan industri, sehingga tercipta kolaborasi yang sangat kuat yang harapannya bisa membawa dampak kepada masyarakat luas dan juga mengembangkan ilmu pengetahuan itu sendiri. Demikian dari saya mengucapkan selamat datang kepada Bapak semuanya di acara International Conference ISAC 2020. Semoga presentasi Bapak Ibu semuanya lancar dan acara ISAC juga lancar, sehingga nantinya selain mendapatkan ilmu yang bermanfaat, Bapak Ibu juga dapat menjalin kolaborasi berkelanjutan untuk kebermanfaatan ilmu kepada masyarakat luas. Demikian dari saya, salam ISAC 2020. Wassalamualaikum warahmatullahi wabarakatuh.
ISAC, Indonesian Science Expo, is the one of the biggest scientific festival in Indonesia. There are many scientific activities in this event, such as conferences, exhibition, and scientific competition, particularly for young people. In ISAC 2019, Econ Probios, one of the member ISAC conference, got many benefits from interacting with various scientific stakeholders in that event. However, now we got different conditions. In ISAC 2020, we cannot enjoy directly, but it held in virtually. So stay healthy in COVID-19 pandemic situation, and you still got benefit from ISAC 2020 by infinity science exploration. Thank you.
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and brongkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, brongkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudek in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of good manufacturing process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
Assalamualaikum warahmatullahi
Finish this session within time. Okay, before we start, I would like to let you know several important points for the audience. First, please have a stable internet connection and make sure that your computer has a working camera and audio device. The chat room is only available for delivering questions to the ongoing discussion in this session. Requesting for changing or moving to the other breakout room in parallel sessions and announcement delivered by the organizing committee. Don't forget fulfill the list of attendance that we will share in the chat room. And if you would like to change the room, please inform our co-hosts and they will assign you to the room of your choice. Okay, it is time to going on to the presentations. For information, we'll have totally 8 presenters, but we will break it down into 2 sessions. The first session will be 4 presenters. Yeah, thank you for attending this session. Okay, for the first presenter, I'll give time to Mrs. Sofa Fajria with the title Antioxidant Compound from Mystica Fatwa How It Lives. For co-host, please play the presentation video.
But I don't think that our findings so far worth telling you.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. First of all, I would like thanks to the organizing committee for giving me opportunity in this conference. I am Sofa Fajria from the Center for Chemistry. Will talk today about antioxidant compound from Mystica Fatwa How It Lives. To start with, I will describe the outline of this presentation, start with introduction, continue to the methodology, research and discussion, conclusion, and the reference of this research. The background of this research are to explore Indonesian natural resources, as long as we know that Indonesia is one of the biggest natural resources. Recently, Back to Nature began around the 19th. The slogan aims to urge us to take advantage of the natural potential that already exists in the world. Unfortunately, about 96% Indonesia still depends on imported raw materials, so that it is important to investigate the of raw material, especially from Mekongas Forest, Southeast Sulawesi, Indonesia. In this research, we explore from Myristica fatwa hot leaves. This plant species originating from India, Borneo, Sulawesi, Maluku, Philippines, New Guinea, Solomon Islands, New Hebrides, Fiji, Samoa, Vanuatu, and New Caledonia. This plant possesses several pharmacological activities such as antioxidant, antibacterial, antiparasitic, and antiproliferative against cancer cells. And the secondary metabolites of this plant mainly are lignan and polyketides such as malabaripin A, malabaripin C, picarin A, and hydroxydenmetodactyloidin. Our primary study from methanol extract from this plant showed that this plant has potency as antioxidant with IC50 26.5 microgram per milliliter. So that the research goals are to isolate and identify methyl 3,4-dihydroxybenzoic MDB from Myristica fatwa leaves, and also to examine antioxidant activity using the pH method, including the compounds from previous research. The methodology of this research, first to literature review, continue to the collecting plant material and determination, extraction and purification guided with antioxidant activity, continue to identify of the isolate using spectroscopy method, and continue to antioxidant examination. From extraction process, we dry the leaves of Myristica fatwa about 1 kilograms, and we got the extract methanol. After that, we continue to vacuum liquid chromatography with hexane, ethyl acetate, and methanol as mobile phase, and we got the extract of hexane, ethyl acetate, and methanol, and we continue to antioxidant activity. From these extracts, we have the ethyl acetate extract show the most active than others, and so that this fraction we continue to the column chromatography, and we got 12 fraction. This fraction we continue to antioxidant assay using the pH, and from antioxidant assay, fraction 5 is the highest activity than others, and continue with separation and purification using column chromatography with normal hexane, ethyl acetate, and methanol as mobile phase, and we got 10 subfraction. Subfraction 3 and subfraction 4 we have already done, and we got new compounds, but these compounds have not examined of antioxidant activity. And then subfraction 5 we continue to separate, and we got compound 3. All compounds will continue to antioxidant assay. Antioxidant assay is using yin and chain methodology. First, the sample we dissolve in methanol to obtain various sample concentration and react with pH. Continue to incubate it for 30 minutes and measure its afterburn at 550 nanometer. And we got the antioxidant activity. The antioxidant activity we calculate the percentage of inhibition. If the color is changed to the yellow, usually the antioxidant is more active than the purple. The IC50 value denotes the concentration of the sample required to scavenge 50% of pH residue. Results and discussion. From this result, we have antioxidant activities from fraction and the extract from Myristica photohotleaves. We can see that the ethyl acetate fraction have the activity highest than normal hexane and methanol fraction. So that we continue the purification of ethyl acetate fraction, and we got 12 fraction. In fraction 5 is the highest antioxidant activity with IC50 16.05 microgram per milliliter. So that the fraction 5 will continue to isolation and purification, and we got the compound 3 and we identified of isolate using FDNMR and LCMS. From LCMS data, we have compound 3 has the molecular weight 168.30. From NMR data, we can see proton NMR that is methoxy in the chemical shift 3.2 and 2, and ABX system rings in the benzene ring. We have ABX system in here, double doublet at chemical shift 7.40 with constant coupling 2.6 and 7.79 hertz that have correlated ortho with proton in the chemical shift 6.8 with constant coupling 7.79 hertz, and have meta coupling at the chemical shift at 7.42 ppm. From carbon spectra, we can see methoxy in here at the chemical shift 42 ppm, and methane at the chemical shift 100 until 115 until 123. That is 3 methane in here, and have 4 quaternary carbon, and in 168 ppm there is characteristics for carbonyl ester. In NMR 2D, we can see the correlation between proton and carbon. In HMQC spectra, we can see the correlation between proton and carbon in one bond. We can see the methoxy have a correlation with methoxy in chemical shift 3.8 with correlation with 42.3, and we can see also in aromatic in here. We can see the chemical shift at 6.8 and with the carbon at 117, and so all the chemical shift from another benzene. From HNC result, we can see the correlation between proton and carbon until 2 and 3 bonded. We can see in here the methoxy have the correlated with carbonyl, and we can see in here, and in benzene rings we can see the correlation between or meta coupling you can see in the HMBC correlation. From the spectra and LCMS spectra, and we compare with literature, that compound 3 have the structure as methyl 3,4-dihydroxybenzoate MDB3. All the compounds, compounds 1, compounds 2, and compounds 3, and we compare the ferric acid as positive control, we can see that MDB has the highest activity than compound 1 and compound 2, but this compound, the ferric acid is highest than as the positive control. Compared with the MDB, we isolate from these plants. The conclusion of this research, first we have isolate 3 compounds that are 2 compounds we from previous research, and the methyl 3,4-dihydroxybenzoate were isolated also from the leaves of Menistica fatuosa. In the antioxidant activities using the pH method, methyl 3,4-dihydroxybenzoate has highest activity with IC50 7.96 microgram per milliliter. This result demonstrated both extracts and isolates are promising candidates as natural resources. These are the reference we use. Thank you very much for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much for the nice presentation from Dr. Sofa Fajriya. Then I will invite the second presenter, Dr. Ahmad Fatoni, with the title Metabolite Detection and Antibacterial Activity of Fungal Endophytic Extracts isolated from Brotowali, Dinospora crispa plants using TLC bioautography assay. For the co-host, please play the video.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, honorable ladies and gentlemen. Thank you very much for your attention. On this occasion, I will discuss and share related to my research entitled Metabolic Detection and Antibacterial Activity of Fungal Endophytic Extracts isolated from Brotowali, Dinospora crispa plants using TLC bioautography assay. The background of this research is related to endophytic fungi that associated with plants. Endophytic microorganisms have their life cycle partly or entirely inside the plants and are located in intra and intercellular spaces or in the vascular tissue. This is the picture of representation showing multifaceted interaction of endophytic with the host plants. The first, reduce herbivore, second, prime of the host plants defensive responses, and abiotic and biotic stress remediator, protection from oxidative burst, and so on. As we know, metabolites have produced by plants and also by endophytes. Metabolites derived from endophyte due to the plant interaction, bilateral synthesis, and plant due to the endophyte induction. The role of endophyte-associated metabolites are defense and competition, bacterial or fungal communication, specific interaction, and nutrient acquisition. Metabolites from endophyte can be used antioxidant, antimicrobial, anti-inflammatory, biofuel, biocontrol, anti-cancerous, and anti-diabetic agents. This is the characteristic of endophyte. They inhabit all the plants, live inside different parts, protect plants from invading pathogens and extreme environmental conditions, and so on. Due to they are capable of synthesizing bioactive metabolites, this study aims to investigate the components of inovative fungal extract and their antibacterial activity using the TLC bioautography assay methods. This is the material and methods. We collected the Dinospora crispa plants from Bogor, Bekasi, and Bandung, and then isolate the endophytic fungi, cultivate in PDB medium, and then extract with the ethyl acetate, and then antibacterial activity using TLC bioautography, and then detect the chromophore on the metabolite in the extract. Metabolite detection using drag and drop for alkaloid. Metabolite detection using foreign culture for phenolic compounds. And then we using phenylene sulfuric acid for staining reagent for obtaining and phenolic. Metabolite detection using cerium sulfate for phenolic and also for amine metabolites. Antibacterial detection using INT in TLC bioautography. This is the table 1, so active fungal isolate associated from stems, petiole, and leaves of Dinospora crispa. And the ethyl acetate extract was obtained from the fungal culture that's grown in potato stachylos plot for 3 weeks. This is the figure of endophytic isolate from petiole leaves and stems of Dinospora crispa. We can see there are 30, 26, and 24 isolate from stem, leaves, and petiole respectively. The study saw that the host plant of Dinospora crispa was taken from Bogor and obtained the most endophytic fungal isolate, probably due to the difference in geographic location and abiotic factors. This is the several endophytic fungi with the distinct exuded colors in the broad media, so the different and diverse fungal species. This is a table 2 on the simple method to detect the secondary metabolite from root extract using TLC and then analyze by using UV lights and spray with staining reagent. We also use the standard of metabolites such as berberine for alkaloid, uroglycinol for phenolic, and quercetin for flavonoid. And we use phenolic compound such as gallic acid for standard. From this figure, we can see several chromatograms of fungal endophyte extract. And we can see from the picture some metabolites such as terpenoid for antibacterial activity. It revealed that several endophytic extracts associated with Dinospora crispa showed potent antibacterial activity. This is the tabulation of TLC dot plot. We can see 6 extracts perform as strong antibacterial. For number 6, number 1, 31, 37, number 8, and 68. This is the figure antibacterial activity using TLC bioautography. The period shown the metabolite have antibacterial activity. We can see for number 28 and 38 of the extract contains flavonoid and coumarin that inhibit the bacterial growth of S. aureus. And we can see from this figure, for number 80, the extra content flavonoid inhibit the bacterial growth of S. aureus. And number 77 and 79, we can see that extract contains coumarin that inhibit the bacterial growth of E. coli. But if we use the TLC, the limitation of the components for compounds with unclear color bands, but these methods provide a fundamental separation and they are segregated isolation. For the conclusion of this research, the study of metabolic detection of the fungal endophytic fungi using TLC.plots can be summarized that 6 endophytic fungi show strong antibacterial activity against both gram-positive and negative bacteria. One extract had excellent antibacterial activity. There is isolate number 6. According to the metabolite using TLC method and with TLC bioautography, the compounds play a role in antibacterial activity, including phenolic, flavonoid, alkaloid, terpenoid. And this study showed that fungal endophytic extract associated with Pinospora crispa may be used promising source of antibacterial. Further study needs to be done to isolate and purify the antibacterial compounds in the active extract, and further investigation antibacterial evaluation by using the microdeletion methods. Thank you very much.
Yeah, thank you very much, Dr. Fratoni, for the nice presentations. So after this presentation, I invite Dr. Abdi Wiraseptama from Research Center for Chemistry, Indonesian Institute of Sciences, to present the presentation. Please co-host to play the presentation.
Good afternoon, ladies and gentlemen. I'm Wakil Presiden from Research Center for Chemistry. Today, I would like to present my research entitled Orthocarpamol, isolated from Orthocarpus heterophyllus, enhances cytotoxic effect of these proteins in H460 and FCF7 cell line. This is the outline of my presentation today: introduction, objective, method, result, discussion, and conclusion. Now let me start to talk about lung cancer. Cancer is the major cause of death all around the world. Lung cancer is one of the most common cancer forms, particularly in men. Lung cancer is unregulated growth in tissue of the lung. On the other hand, breast cancer, it has been known that this cancer is the most common type of cancer found in humans and causes high rate of mortality and morbidity. Conventional cancer therapy methods, including surgery, radiotherapy, and chemotherapy, can offer major curative effects against localized tumor. However, the use of these therapies may cause the incidence of tumor recurrence and lead to development of any malignancy as well as drug resistance. Therefore, we need to find alternative strategy to overcome this problem. One of the best strategies is the use of combination drugs with natural compounds that boost anti-cancer activity, and this combination leads to the synergistic interaction. Natural products always become potential source for discovering anti-cancer activity. It is because natural products provide several benefits, for example, less side effects, more efficacy, safety, better compatibility, and cultural acceptability. Orthocarpus heterophyllus or jackfruit has been used in traditional medicine in many countries. including Indonesia. This plant contains several flavonoids complete interaction of this compound. So the objective of this study is to investigate the interaction of autocarbonol in combination with cisplatin against two cancer cell lines, including non-small lung cancer and breast cancer. First step is determination of cytotoxic activity. In this study, cell suspension was split in the 96-well plate and then incubated for 24 hours. After that, compound cisplatin with 5 serial concentration were added in the plate and then incubated for 72 hours. After incubation, 20 microliter of MTT was added into the plate and then incubated for 4 hours. After incubated and aspirated the media, 50 microliter of DMSO was added in the plate and then read the optical density using microplate readers at 570 nanometer, and percentage inhibition was calculated using this formula. The results show that autocarbonol exhibit cytotoxic effect against non-small lung cancer cells with IC50 value 16.88. While in comparison with cisplatin, where cisplatin has the IC50 value on non-small lung cancer, about 12 carbonol has cytotoxic effects against P16 melanoma cell line. So from this result, we interested to know the interaction between autocarbonol and cisplatin in combinations. Again, two kinds of cell lines, including non-small lung cancer as well as breast cancer. So next study, we use modification of MTT to know the synergistic effect of autocarbonol and cisplatin. In this study, cell suspension was split in the 96-well plate and then add combination of autocarbonol and cisplatin. Then incubate for 24 hours. After incubation, 20 microliter of MTT was added into the plate and then incubate for 4 hours. After incubation, 100 microliter of DMSO was added into the plate, and then the optical density at 570 nanometer, and combination index was calculated using this formula, where the combination index value was defined as synergy if CI less than 1 and additive if CI equal to 1 and antagonist if CI more than 1. This is the result of interaction between autocarbonol and cisplatin. We can see here that the combination of autocarbonol and cisplatin have the IC50 value was below the stretch line, and CI index of this combination is around 0.09. That means that the combination of cisplatin and autocarbonol have a synergistic interaction against non-small lung cancer cell. In case of MCL7, it can be seen that IC50 value of autocarbonol and cisplatin was below the stretch line, and the CI value of this combination was around 0.13 to 6. This means that combination of cisplatin and autocarbonol have the synergistic interaction. In order to know the effect of this combination on the morphological change of the cell line, we use fluorescent microscope to know to observe the change of the morphological of the non-small lung cancer as well as breast cancer. In this study, the cell suspension was split on the microplate and then a combination of autocarbonol and cisplatin. Then incubate for 24 hours. After incubation, combination of AOPI and PI, acridine orange, and propionate iodide were added into the plate, and then observed under fluorescent microscope. And the results show that there was a change in morphological, both cell line, non-small lung cancer as well as MCF7. See that the combination of octocarpanol and cisplatin led the change in the morphological on non-small lung cancer as well as MCF7. It has been known that dual action of dual standing AONPI was relatively stable of 4 image-based variability of analysis. The result exhibit that the treatment with combination of octocarpanol, the number of cells was lower than treatment with single compound. The morphological observation after treatment with the mixture octocarpanol and cisplatin for 72 hours demonstrated that the combination led to the significant change in apoptotic cell and some membrane bleeding compared to treatment with single compound. And the conclusion of the study, octocarpanol may enhance cytotoxic effect of cisplatin and synergistic interaction. This combination only requires low concentration that afford to therapeutic dose against tested cancer cell line, including H-positive non-small lung cancer line as well as MCF7 cell line. The result of the study proposed the potential of octocarpanol as adjuvant of cisplatin in vitro. We would like to thank for the sponsor and thank you very much for your kind attention.
Yeah, thank you very much for Dr. Abdi for the nice presentation. And then we'll continue to the next presenter. I invite Dr. Arthur Alalono from Research Center for Chemistry, Indonesian Institute of Sciences. Please, co-host, to play the video.
Good afternoon, ladies and gentlemen. My name is Arthur. I would like to present our current research about the extractions and identification of nitrogenine from the kratom leaves. Kratom, it is a plant known to Indonesia, indigenous to Indonesia, especially from the Borneo Island. It's been used in traditional medicine. In one several properties that give a distinctive about the kratom, it has a high similar to that opiates and opiates from the opium. So this makes the kratom itself is very unique and very, in some point, very controversial. The kratom effects have 3 colors, leaves. I mean, they have the green, white, and red. This different gives a very different use and different properties. Many people in Indonesia, especially in the traditional way, they use kratom for the natural stimulants. It gives a quick energy boost and relieves pains naturally. In some research also reported that it helps treating diabetes. The substance, like I said before, opium-like substance in kratom, it gives us stress reliefs, and in some area it also have the use in the rehabilitation of narcotics victims. In some point also it helps the sleeping for those who have sleeping problems. Usually they use it in the traditional way in vision in teeth. I mentioned before, kratom has a very polemic in the National Agency of Drugs and Narcotics indicates that they're suggesting in 2022 kratom should be registered as the first class of narcotics plant, and some point, yes, because they have the indications of the addictive and also can lead into the death. But we know that kratom in the Borneo Island is many farmers depending on this kratom leaf for their economic. What they're doing now, they're collecting the raw and the dry leaves, and they just chop it in this traditional way, and they dry it and they sell it as a product, even export it to countries outside Indonesia. For this reason, the two contradictive areas in one point that researchers being challenged, especially here from the governor of the West Kalimantan, to give a good explanation result about this kratom, the use, the positive area. In some point, the discussions about the kratom itself in one point of their use, the traditional use, the medical use, and also for their economic, and also even the possibility of miscarriage in the narcotics area. In here, we can see that there's several products of kratom leaves being sold in Kalimantan. Even there, to support this idea, the program in the Kalimantan, they're putting the kratom study is to support the economic society which really relies on this kratom leaf for their basic needs. What makes kratom so special? I mean, what is so special? Because in kratom itself, the leaves especially, it consists of nitrogenin and 7-hydroxynitrogenin. These two substances, or these two compounds, are very distinctive in kratom, such as cinchonin or cinchona in quina leaves, or artemisinin in artemisia. Nitrogenin, and together with 7-hydroxynitrogenin, is an alkaloid substance which gives sedative and analgesic properties. Nitrogenin itself is around 66% of the alkaloid content, so it's the primary one. And it gives several activities that gives kratom leaves an advantage towards the others. Since an alkaloid, so the conventional way to extract it is using the alkaloid extraction method. So we acidify it, we give additions of, we increase the pH level. So we're using the acid-base reaction to obtain the crystalline product of the alkaloid. It is time-consuming, and it's a very chemical. Lots of chemicals being used. We're shifting it into the much more extraction way, which is much more greener way, using the C134A. Why are we using that? Because we've been using it also for the extraction of artemisinin inside from the artemisia using this kind of method. It's very precise, it's very selective, 28 times than the conventional one, 45 times faster. So basically, it is very selective. We also conducted using the tobacco, and we have a result showing that it has a selectivity also in nicotine. So since nicotine is also an alkaloid, why don't we try using this method inside to obtain the in kratom? So 500 grams is being used. We put it in the chamber, extraction chamber, reaction for let's say 1 hour, and one atmospheres, and 25 degrees room temperature with 3 times cycles. So the extraction puts in the extraction chamber, and then we put the HFC, which is we're acting as the solvent here, into the collector. Then we manipulate the temperatures and the pressure there. So the freon, the HFC134L, will be converted into the gas, while the extract will be remain in the chamber itself. So those gas cycles go on. So it's a closed system. So therefore, it will be much more effective. Why it's so easier? Because the property of the HFC134A has a boiling point at minus 26 degrees Celsius, meaning that at room temperature they'll be gone. They'll be changed from the liquid into the gas. So this is the GTMS of the kratom extract using the FC134. We can see here by the analysis that the 18.3 minutes peak consists of the mitragynine, comparing from the database. We want to confirm again here using the LTMS. The blue peak here indicates several peaks of the extract itself, showing that 7.1 minute and 7.52 minutes on the LCMS chromatogram of the HV kratom extract indicates that compound is mitragynine and 7-hydroxymitragynine comparing to the LCMS database. We want to confirm that, checking the mass spectrum also from the 7.12 minutes, it shows that it gives the MH plus at 399.22, which also consists of the mitragynine. So from this result, that we can know that from the extraction of dry leaf kratom using the FC134A, the extract consists the targeted compound, which is the 7-hydroxymitragynine and mitragynine. The homework for us is now that we have to further work how to isolate this peak at 7.12 and 7.52 minutes to obtain the crystalline product of the targeted compound, which is hydroxymitragynine and mitragynine itself. In other addition, we would like to identify other products as possibility other secondary metabolites also being extracted. So for the conclusion here, is that the extraction of kratom leaf using the FC134A giving a 0.15% from the dry leaves. We have to calculate again how much the amounts of the mitragynine after the isolation from the extract itself, which is at the 7.12 minutes. And the GCMS and LCMS retention time indicates the presence of mitragynine in the kratom FC134 extract with a fragment of 399.22. So it is a very good subcritical system to extract the selected, especially in our case here, it is mitragynine and hydroxymitragynine. And this is the first report of extraction of mitragynine from the kratom using subcritical system like artemisinin. Later on, this can be used for further production or isolation of mitragynine from kratom. Thank you very much for your time. I hope we have a very fruitful discussion. Thank you.
Yeah, thank you very much for Dr. Arthur for the very nice presentations. What a wonderful presentation from all of the presenters in these sections. I can say this section is a special section for expert from Research Center for Chemistry. So because of the section, all the presenters come from Research Institute. Indeed, there are several questions for the presenters. Today here, I would like to read the questions from the audience. The first question coming from Muharni from Unsri to Ahmad. What is possible to search for bioactive compounds from endocritic microbial biomass? Which one is more effective than the medium? Please, thank you, Dr. Ahmad.
Thank you for the question for me. In my opinion, we can use the biomass and also the broad media for to get the bioactive compound of the endophytic fungi cultures. We in preliminary study also usually use both broad and also broad media and also biomass to get the bioactive compounds. And which one is more the effective? I think we must check the difference metabolite from the biomass and also from the broad media to confirm the bioactive compound, and we can spirit the bioactive compound with maybe chromatography, column chromatography, and also with HPLC preparative, I think. Yes. Okay, maybe it can be answer your question. Yeah.
Thank you, Dr. Ahmad. So how about the Miss Muharni? Is there any question? Please direct to Dr. Ahmad.
Yeah.
Okay, clear. Thank you.
Okay, clear. Yeah, thank you very much. We are going to the second questions coming from Elvita from Unsri as well. To Dr. Sofa Fajriah, how does the antioxidant activity of the methanol extract compare to its pure compounds? Is there a possibility that the antioxidant activity will increase if the compounds with moderate activity are combined? Yeah, please, Dr. Sofa Fajriah.
Thank you, Pak Andi. Thank you, Ibu Elvita, for the question. Maybe I will answer. From our result that pure compounds more active than methanol extracts. Pure compound, it means 3-methyl 3,4 dihydroxybenzoate is highest activity than methanol extract, but methanol extract because still extract below 200 microgram per milliliter still high for extracts to get antioxidant like that. And is there a possibility maybe if we can increase to compounds with moderate activity if combine the compounds, but we have to using the examination of what the properties of the compound itself, and then we can using in silico study first to see is it these two compounds can increase or maybe will decrease if we combine the compounds. Thank you.
Yeah, thank you, Dr. Sofa Fajriah, for the answer. So, is there any comment from Elvita to respond the answer, please? Oh, I think it's already very clear. Yeah, thank you very much. There are also another questions coming from Bapak Gunawan Pasaribu from P3HS, P3HH to Bu Sofa, Bu Sofa become idola today. Do you have any comparing data IC50?
Thank you.
With Mictica fragrance?
Okay, thank you for the question, Pak Gunawan. Myristica fragrance is formerly leaves of Myristica pala. We can compare with the literature data. Many literature show that the activity of Myristica fragrance. In here I see in the journal Myristica fragrance in the ethanol extract have activity about 30 antioxidant activities, so that maybe not really far in the antioxidant activity which I have. Myristica fatua has 26 microgram per milliliter, but I think not really far for the activity. And I have a literature review for this Myristica. They're also similar with the secondary metabolite of this compound, but for Myristica fatua still not many literature to explore of this plant. Thank you.
Okay, thank you.
Okay, thank you. It's already seems clear for Bapak Gunawan Pasaribu. Yeah, still there are many questions. So the next question coming from Abdi Lipi, Dr. Abdi Lipi to Dr. Arthur. in LIPI as well, or maybe both of you just sitting next together each other. Okay, the question is, I would like to ask you, since we know mitragynine has strong effect in CNS that has mimic morphine effect. How is the regulation, the use of this plant and derivative in Indonesia? Yeah, please, Dr. Arthur.
Yeah, thank you very much, moderator, Dr. Andi. Yeah. Regarding this area, yeah, there's conservations of this kratom leaf itself. It is yet not been registered as the first drug plants. Therefore, the Ministry of Health of Indonesia are giving an effort to make scientific evidence a good, reliable, showing that this kratom, it is actually has a lot of medicinal use. And then we like to prove in this our study here that using the subcritical system, we can isolate and extract the mitragynine, which is the like cannabidiol in cannabis, which is the main problem. Let's say that we can extract the pure compound and sold it as the pharmaceutical API rather than sell the leaf itself for the misuse of this plant itself. Yes, I have to admit this plant is still in conversation. BNN, the narcotic agency, would like to put them in the first category of the narcotics, but yet there is still debate between these two institutions. So therefore, we have to give more evidence because it gives lots of economic values, especially for the West Borneo and the Borneo island for the society. That's my explanations currently. Thank you.
Yeah, thank you for the explanation. Is there any comment from Dr. Abdi?
Thank you, Dr. Arthur, for your comment. Yes, interesting indeed, because if we talk about this plant, it's quite similar with marijuana, right? Yes. Cannabis, because like that, just like marijuana. You know that in my college, my former college in Thailand, also work in this area, in this plant, they're using the micropropagations to enhance the production of nitrogen. But in Thailand itself, it's not the prohibited plant actually, just like marijuana. Thank you, Dr. Arthur.
Thank you.
Yeah. Okay. Thank you. And we go to the next questions from Elvita, from Unsri to Pak Fatoni, Pak Ahmad Fatoni. Are there difference in chemical content, qualitative and quantitative, of the same endophytic fungi of Brotowali collected from different locations? And then why happen? Thank you. Please, Dr. Ahmad Fatoni.
Okay. Thank you, moderator and Ms. Elvita for the answer, for the question. In my research, we collect the endophytic fungi from Brotowali in different location, but the qualitative analysis not yet do in my research, but not quantitative but qualitative analysis. And chemical content in the endophytic fungi in my research is a little bit different, not only because the different location collect, but also that is maybe because the biotic and abiotic contribution for the metabolic production, and also the difference of the isolate we collect. Maybe that is can contribute to the different chemical contents. And in my research, we just the qualitative in TLC chromatogram, and so in quantitative we must confirm by instrument analysis and also other. Yes. Thank you.
Yeah, thank you, Dr. Fatoni. Is there any comment from Bu Elvita of Dr. Fatoni explained?
Yes, thank you.
Okay, thank you. Yeah, there's still one more question from Nino from LIPI to Dr. Arthur. It is nice and interesting to presentation about the kratom extract. So far, I know the kratom is still prohibited in Indonesia. Is it true? What is your opinion about the situation if kratom is a lot benefit for health but is still not allowed? How about other plants such as marijuana? The active compound is same with kratom. Thank you. I think this is more and less similar to Dr. Abdi comment or Dr. Abdi question. Yeah, but please, yeah, Dr. Arthur.
Yeah,
thank you for response.
Thank you very much, Pak Nino, for your questions. I'd like to give an explanation. Just now, today, from 10 until 1, there is an FGD between the Badan POM and the Ministry of Health regarding this plan also, whether they want to put it as to support as a medical plans or they want to retract it and give it to the narcotic agency to be put as a narcotic plans in the first category. Now it's still on debate, like I said, mentioned before. Until now, the regulation said has not mentioned that kratom is inside the category as the what do you call it, narcotic plants. But yes, the substance like mitragynine has 10 times more effective compared to opium. This is very, very, very interesting because it's very high activity in suppressing the nervous system. But it has a beneficial. For example, we can take this mitragynine, isolate this, and convert it to API, and then can be used in the pharmaceutical area. It will give lots of benefits. For example, this extraction method also can take cannabidiol from cannabis using the subcritical system. So, in my opinion, I have discussion with the BNN. My colleague says whether if you have to burn all those marijuana being seized on the market with no values, why don't you just take the cannabidiol or the mitragynine, take it as the API and put it in the pharmaceutical values for precautions or maybe in what do you call that, the rehabilitations. They need those stuff in the point of pharmaceutical use, not in miscarriage use as a drug addiction, but more compared to that. It has a very high value. So therefore, we would like to offer this technology to isolate the kratom for now.
Oh, I think enough. There is no response from Bu Elvita. Oh, I'm sorry, from Pak Nino. Is there any comment from Pak Nino?
Yeah, because yeah, thank you, Dr. Arthur. Yeah, this is very interesting because as we know that kratom is, I mean, have a high value in health also, and also in as an import substance in America, right? So if we can use this for the health, is very useful for human. But yeah, just like Dr. Arthur said, we still have a problem with law enforcement. Stuff like this. Well, I think it's thank you, Dr.
Thank you.
Yeah, thank you very much for the presenters, as well as the question for these sections. However, we still have another 4 presenters. So time wisely, we will go to another sessions. But before we go to another this coming section, yeah, I would like to thanks to the 4 first presenters and give applause to them. Okay, we are going to the next sessions. Here I will invite 4 presenters for the next presenter. First presenter will be Dr. Evita. Sorry, are you attending this I think you're already here, right? Dr. Cici Darcy. Hello, Dr. Cici Darcy.
Yeah.
Okay, and Ibu Septi Nurhayati.
Yeah.
Okay, and the last one is Dr. Fitri Setioningrum. Okay, I think all the presenters already attend this room or this section. So I invite Dr. Alvita from Dr. Alvita coming from please to the co-host to play the video.
Assalamualaikum warahmatullahi wabarakatuh. I am Alvita from Sriwijaya University. Thank you for this opportunity. The title of my presentation is Antioxidant activity of endophytic fungi extracted from the stem bark of Swietenia mahagoni. Our research team is Elvita, Hani, and Yohanini, and Fadila. Swietenia mahagoni, known as mahogany, is a medicinal plant that has been used to treat various diseases such as to prevent degenerative disease. The host plants that are used as drug with antioxidant effect are candidate for treating endophytic fungi that produce antioxidant activity. To obtain antioxidant compounds, this research step is established to obtain the antioxidant active extract. Next step is isolation of the co-compound, identification of the structure, activity test, 2-year antioxidant compounds. This figure 2, so isolation and endophytic fungi from in PDM media. Obtained to 7 strain endophytic fungi. In previous studies, we have reported the isolation of endophytic fungi from the leaf stalk mahogany and their antioxidant activity. In the previous study to obtain one compound not antioxidant activity, and the previous study in the present study, the study I made to isolate the antioxidant compound from the Cordosporium tenacinium. Two part of the host, left stalk and the stem bark, extract to obtain equivalent antioxidant activity. DMG3 for the left stalk mahogany and DMG4 from the stem bark with equivalent antioxidant activity. The fungus was identified with molecular method. The similar by analysis phylogenetic, the fungus is Cordosporium tenacinium. Then bank association number. This the association number in Bank IMN 379836. This figure, the stack of isolation of two compounds from endophytic fungi, Cordosporium tenacinium, from the stem bark of the mahogany, blue, were compared with the state of previous study of the Sampungus from the left up mahogany in red. The table 2, I see 50 value extra of 2 compounds from endophytic fungi from staff left staff and from stem staff display so to table 2 extra IC50 equivalent for left staff compound 1 active good antioxidant activity. The compound to obtain previous study is not active antioxidant and standard positive control to antioxidant activity ascorbic acid. The spectrum of compound 1 to NMR proton spectrum indicate the aromatic proton, 2 aromatic proton with multi-crystal doublet, 1 methoxy proton, 1 methyl proton, 1 methylene proton. This is the NMR carbon. The spectrum shows the 10 carbon, 6 aromatic carbon signal, and the signal is carbon keton, and the signal carbon carboxylate. 2 signal is oxyaryl carbon. The signal is methoxyl carbon. Based on the analysis spectra, NMR proton and carbon, the compound 1 was identified as phenolic derivative with 4 subsequent consisting of hydroxyl, methoxyl, methyl keton, and acetyl group. The figure 6, the structure of phenolic derivative from corresponding penicillin. 2 compound was extracted from the fungal. One compound is inactive antioxidant and 1 compound good antioxidant activity. The inactive antioxidant, the absorption of the proton by the PPH free radical produce new free radicals that are less stable. The presence of heteroatoms in the ring stem to inhibit free radical response in microbe. And compound 1, good antioxidant activity, the absorption of the proton by DPPH free radical is much more comfortable and can form new free radicals that established by distribution and develop the localization electron in the molecule. Conclusion: the endophytic fungi Berkepribadian penicillin has been found in the stem part and the leaf part of mahogany. However, the chemical content are the same. These studies of the Berkepribadian penicillin could be used as a new source of antioxidant. So the research is needed to isolate the alternative compounds in Berkepribadian penicillin. Thank you.
Yeah, thank you very much. much for Dr. Alvita from Sriwijaya University. And then for the next presenter, I would like to invite Dr. Cici Darsi from Research Division for Natural Product Technology, Indonesian Institute of Sciences. Yep, please co-host to play the video.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon. Today I would like to present our research about in vitro antioxidant activity of macroalgae Sargassum duplicatum and Palmaria palmata extract collected from sepanjang page Gunung Kidul, Yogyakarta. We are from Research Unit for Natural Product Technology, Indonesian Institute of Sciences. Introduction. Marine macroalgae are considered as important sources for bioactive phytochemical with promising application in drugs, cosmetic, and food. Macroalgae were classified into four major groups. They are green algae, Chlorophyceae, red algae, Rhodophyceae. Moreover, macroalgae also contain vitamin, protein, inorganic element, and carbohydrate. Sargassum duplicatum belong to green algae. Some research reported that the metabolite from sargassum show various pharmacological activities such as antioxidant, antibacterial, anti-inflammatory, anti-cancer, and tyrosine inhibition. And for Palmaria palmata, this macroalgae is classified into red algae and also known as terus. Some research reported that metabolite from this algae exhibit biological activities such as antioxidant, angiotensin-converting enzyme inhibition, anti-inflammatory, and inhibit primary human neutrophil activation. In Gunung Kidul, Palmaria palmata traditionally consumed as snack food. People in Gunung Kidul fried this macroalgae and consumed directly. Whereas Sargassum duplicatum has not yet widely used by the Gunung Kidul community. So why the aim of this study was to determine the total phenolic content of extract from Sargassum duplicatum and Palmaria palmata. And we want to know the biological activity of this drug, especially for the antioxidant of Sargassum duplicatum and Palmaria palmata extract. Methodology, we collected the Sargassum duplicatum and Palmaria palmata from sepanjang page Gunung Kidul, Yogyakarta, Indonesia. The sample were then identified in Plant Systematic Laboratory, Biology Faculty, Gajah Mada University. The macroalgae were then dry at room temperature for 7 days. For the extraction of sample, the macroalgae were macerated using 3 solvents. They are chloroform, ethyl acetate, and methanol at room temperature for 24 hours. The crude extract of these macroalgae were obtained by evaporated filtrate using rotary evaporator. And for the total phenolic content of Sargassum duplicatum and Palmaria palmata extract were determined using Pauling-Keough record. And for antioxidant activity of this extract were determined by GPPH method. Result and discussion. The results show that the yield of methanol extract from Sargassum duplicatum and Palmaria palmata were higher than other extracts. The polar solvent methanol was effective to extract the compound contained in the sample. And for the total polyphenol content, The results show that the TPC of ethyl acetate extract from both macroalgae higher than other extracts. Total phenolic of ethyl acetate extract from Sargassum duplicatum higher than Palmaria palmata with a value of 726.54 mg. And this study was an agreement to the previous research. The phenolic acid and quercetin were confirmed in ethyl acetate extract of Sargassum plagiophyllum. And for the antioxidant activity of extract, the result reported that the antioxidant activity of ethyl acetate extract from Sargassum duplicatum and Palmaria palmata were similar. The activity from both macroalgae around 700. This antioxidant higher than chloroform and methanol extract. The scavenging activity of the Sargassum duplicatum and Palmaria palmata extract related to TPC. The results were similar to the previous research reported that TPC and antioxidant activity on brown algae higher than green and red algae. For the conclusion, first, the yield and TPC on Sargassum duplicatum and Palmaria palmata extract were affected by the polarity of solvent. Second, chloroform, ethyl acetate, and methanol extract of Sargassum duplicatum and Palmaria palmata show inactive antioxidant properties toward TPPH radical with the activity more than 250 microgram per milliliter. It is suggested that the other biological activities would be conducted on this extract. Thank you for the attention. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you for the interesting presentation from Dr. Cici Darsi from Yogyakarta. So I will invite the next presenter today, Dr. Septi Nurhayati from Yogyakarta as well, from Research Division for Natural Product Technology, Indonesian Institute of Sciences. Yeah, please, the host to play the video presentation.
Assalamualaikum warahmatullahi wabarakatuh. My name is Septi Nurhayati from Research Unit for Natural Products Technology, Indonesian Institute of Sciences. What I'm going to present is actually still only in its early stages, but I really think that our findings so far worth telling you. It's about phytochemical properties and antioxidant activity of wild-grown and cultivated Ganoderma rhizobium. I will begin with the introduction to Ganoderma rhizobium. Ganoderma rhizobium, macrofungi, commonly known as lindsay, has been used as a therapy for the prevention and treatment of various diseases for centuries in Asia. The mushroom has anti-tumor, hepatoprotective, anti-inflammatory effect, and so activities in the immune. Most of these therapeutic activities are believed to have association with its antioxidant activity. Due to their different growing conditions, the wild-grown and cultivated Ganoderma rhizobium may contain different levels of effective chemical components, which may affect their quality and medicinal efficacy. The present study was carried out to determine the differences between wild and cultivated Ganoderma rhizobium, which might be useful in exploring the characteristic of the chemical compound of Ganoderma rhizobium regarding its antioxidant activity. The material effort. Mushroom materials, wild-grown Ganoderma rhizobium were collected from Cianjur and Gunung Kidul, and the cultivated were collected from Kaliurang, Godean, and Gunung Kidul. Extraction method: samples were macerated in ethanol 70%, 1 in 15 at room temperature for 72 hours. And the physicochemical evaluation using gravimetric method include total S content, acid insoluble S, water soluble extractive, ethanol soluble extractive. And the phytochemical evaluation: the first, water-soluble polysaccharides using phenol sulfuric acid method, and the total phenolic content using polymethyl reagent, and the total terpenoid using method of Lin et al. Antioxidant activity was evaluated by using radical scavenging activity using diphenyl dihydrazine DPPH pre-radical assay, and the quercetin was used as a positive control. The antioxidants activities presented as IC50 values. The result and discussion. The first, the result of physicochemical evaluation. As you can see here in the table, the highest and the lowest total S value were found in cultivated Kaliurang and in cultivated Godean respectively, while the highest and the lowest acid insoluble S value were found in cultivated Kaliurang and in wild ground Cianjur respectively. The highest water soluble and ethanol extractive value were found in cultivated Kaliurang, whereas the lowest water soluble and ethanol extractive value were found in wild Gunung Kidul and in wild ground Cianjur. Even though wild ground Gunung Kidul has the lowest water soluble extractive value, it has higher ethanol soluble extractive value compared to cultivated Gunung Kidul and cultivated Godean. Total S value provide an overview of inorganic composition and other impurities in dried mushroom as content parameters related to the purity and contamination. The acid insoluble S value indicates the presence of silicaceous substances. The determination of extractable matters refers to the amount of constitution in material which is extracted by specific solvent. Polar compounds tend to be more attracted to and are more soluble in polar solvents. It is indicating the nature of constituent of the raw materials and also help in detecting low-grade material. Water-soluble and ethanol-soluble extractive values plays an important role in evaluation of group drugs. Less extractive value indicate addition of exhausted material, adulteration, or incorrect processing. The result of phytochemical evaluation: the water polysaccharides were the most abundant metabolite compound in Ganoderma lucidum, followed by terpenoid and phenolic based on the sample. Cultivated Ganoderma lucidum from Godean has the highest water-soluble polysaccharides and phenolic content among other studied samples. Whereas cultivated Ganoderma lucidum from Gunung Kidul has the lowest water-soluble polysaccharides and phenolic content. Both of wild Ganoderma lucidum have higher terpenoids content compared to all of cultivated. Wild-grown Ganoderma lucidum are grown spontaneously in self-maintaining population in natural or semi-natural ecosystems and can exist independently of direct human action. Cultivated Ganoderma lucidum have arisen to human action and are grown for their use in mushroom farm. Metabolites in mushroom could be affected by physical variation, ecological condition, terrestrial variation, genetic genetic factor and evolution. Another research showed that production of metabolites was influenced by environmental factors such as humidity, temperature, intensity of light, supply waters, mineral, and carbon dioxide. The result of antioxidant activity, the scavenging capacities of well-grown and cultivated Ganoderma lucidum range between 344.15 and 688.05 micrograms per milliliter. Quercetin was used as a positive and exhibited IC50 value of 10.55 microgram per milliliter. The antioxidant activity of Ganoderma lucidum decreased in older cultivated Korean and then wild Gunung Kidul, wild Cianjur, cultivated Kaliurang, and the last cultivated Gunung Kidul. The cultivated Korean revealed the highest DPPH scavenging activity among the studied samples. Both of wild brown Ganoderma lucidum have higher DPPH scavenging activity than a sample from cultivated. Okay, and the conclusion, the present investigation demonstrated that the physicochemical, and antioxidant properties of wild brown and cultivated Ganoderma lucidum were significantly different. Cultivated Ganoderma lucidum from Godean has the highest water-soluble polysaccharides and phenol content. Both of wild brown Ganoderma lucidum have higher terpenoid content compared to all of cultivated Ganoderma lucidum. The cultivated Korean revealed the highest DPPH scavenging activity among of the studied sample. This research was supported by National Priorities Program, Indonesian Institute of Science. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much for Dr. Septi Nurhayati from Yogyakarta for the nice presentations. And then we'll continue to the last but not least presenters today. We have Dr. Fitri Stioningrum from Research Center for Biotechnology, Indonesian Institute of Sciences. Yeah, please go host to play the presentation video.
Thank you very much. I would like to present our research antibacterial activity of solar garlic. This research was conducted by myself, Fitri Stioningrum, Gunawan Priadi, Vivi Afiati, Nina Herlina. We are researchers of Research Center for Biotechnology, Indonesian Institute of Sciences. Well, Indonesia is well known around the world about its biodiversity, including plants, species, or herbs. For centuries, local people using herbs or species for recovering of illness or against of disease. Why? Because local people believe that there are bioactive compounds that can secure their health. Several researchers report that herbs or herbal have antibacterial effects or antibacterial compound. There are tannin, flavonoid, phenolic, alkaloid, saponin. In our research, we choose solo garlic to investigate the antibacterial activity. Why? We choose solo garlic because comparing with multi-clove garlic, solo garlic has higher alicinous components, so it influences the antioxidant capacity more higher than multi-clove garlic. Aging solo garlic become black garlic could produce or increase as allyl-cysteine as antioxidant component 5 to 6 times higher, and then could increase phenolic content 5 to 8 times and decrease fructan content. In our research, the aim was investigate antibacterial activity solo garlic either in fresh from or black garlic solvent. We choose two solvents, isopropanol and ethyl acetate. We use two methods of antibacterial activity. There were diffusion and dilution methods, and we used two pathogen bacteria. There were E. coli and Staphylococcus aureus. For main material, it was solo black garlic we obtained from local market in Bogor, and we use pathogen bacteria of ATCC strain, and we use another analytical reagent and chemical for analysis. Our method was divided to several steps. First step was fermentation of solo garlic for 7, 14, and 21 days, and then we extract the active component by maceration method, and then we prepare tested bacteria. We also measure of flavonoid, and the main method is antibacterial activity by diffusion and dilution method. This is our result. In this table, we can see that black garlic fermented in 4 weeks has higher total phenolic content other treatment, and isopropanol extract has higher flavonoid content than ethyl acetate extract. Why? PG4 has higher phenolic content other treatment because it was supported by Ian Liu and Choi et al. That heating has large influence on flavonoid availability, and flavonoid content of multi-clove black garlic increased significantly up to 21 days of aging. This is the table of antibacterial activity by diffusion method. PG42 in isopropanol extract and can against higher E. coli and Staphylococcus aureus than positive control. This is a table of minimum inhibitory concentration of BG4. We use BG4 for dilution method because BG4 has higher antibacterial activity by diffusion method. We can see that MIC value of PG4 against E. coli was equal. It was the acetate. Both of extract solvent of PG4 had the MIC level against E. coli 1 milligram per milliliter. Otherwise, isopropanol extract of PG4 saw lower MIC against Staphylococcus aureus than ethyl acetate. Thank you for the time. We would like also to Ministry of Research, Technology and Higher Education of Republic of Indonesia through INSINES program. Thank you very much.
Yeah, thank you so much for Dr. Chris joining room from ISAC Center for Biotechnology for the nice presentation. Yeah, ladies and gentlemen, we still have around 15 minutes to discussions. So here in chat room, we already get several questions. The first question is coming from Dr. Elvita, from Unsri to Dr. Cici Darsi. Yeah, this sample has inactive antioxidants. Whether these results have been compared with the same sample from the literature, maybe the question is about the comparison with the information from the literature. Yeah, please, Dr. Cici Darsi, to give a response.
Thank you, Pak Andi. Thank you for the question, Ibu Elvita. I will try to answer the question. Actually, we already compared the result with another literature, and for another literature reported that the efficacy less than 250 microgram per milliliter. But I'm sorry, I forgot the exactly for the value. But for the efficacy for another literature is active. Thank you.
Yeah, thank you.
Maybe that is my answer.
Thank you.
Dr. Elvita, do you feel enough with the explanations? Oh, yeah, okay. Okay, to the next questions coming from Dr. Muharni from Unsri as well to Ibu Septi. Why there's such big difference in antioxidant activity, but the chemical content of phenolic and flavonoid is not relatively different? Whether there are what other factors that determine antioxidant activity? Yeah, I think it's already clear question to Ibu Septi, please.
Okay, thank you for the question from Ibu Muharni. Maybe you mean polysaccharide. Yeah, we only evaluate polysaccharide, terpenoid, and polyphenol. We didn't evaluate. We don't evaluate flavonoid. And why the value of antioxidant activity different? I think because the bioactivity, especially antioxidant, was believed to be associated with different content of many bioactive, and I think it's not only depend on polysaccharide, polyphenol, or terpenoid, because it's still only it's still there are a lot of many bioactive that contain in Ganoderma lucidum, and if we based on our study that the highest DBPH scavenging activity it's revealed that the highest DBPH scavenging activity it's cultivated godean, and the cultivated godean has the highest water-soluble polysaccharide. It means maybe that there is a correlation between water-soluble polysaccharide and DBPH scavenging activity, but it still need to be confirmed, maybe in further study. Thank you.
Yeah, thank you, Dr. Septi. And please, if you don't mind, to open your camera during this section, so we can get each other, contact each other better. Yeah. The next question. Still, we have any other questions? The next question is coming from Dr. Abdi from LIPI to Dr. Fitri. Recently, substance that can be considered as active antibacterial agents is a substance that has MIC value lower than 10 microgram per milli. In this study, did you use broad microdilation method? Yeah, please, Dr. Fitri.
Thank you, Dr. Abdi. In our study, we didn't use microdilution method, but the lower concentration in our study, it was 0.25 milligram and milliliter, but it didn't show a good antibacterial against Staphylococcus aureus or E. coli. And other question? Oh, I'm sorry.
Oh yeah, thank you, Dr. Fitri.
Okay, thank you.
Get any comment? Oh, thank you, Dr. Abdi. Yeah, we're going to the next coming question. The next coming question is coming from Dr. Sofa from LIPI to Ibu Fitri. Still to Ibu Fitri. What is the correlation between TPC, TFC, and antibacterial activity that you use in this study? Yeah, please give some.
Okay, thank you, Dr. Sofa. It is. Did you mean TPC is total phenolic content and TVC is total flavonoid content?
Yes, yes.
Yeah.
We don't measure about the TPC, but we measure total flavonoid content that it correlates with antibacterial activity, because higher total flavonoid content in the extract that produce higher activity bacterial. But we don't measure, or we don't investigate what's the flavonoid compound in solo garlic or solo black garlic, because we didn't have the standard compound.
Maybe you can compare with the literature. Is there maybe some flavonoid in there?
Yes. Several literature. Unfortunately, there are limited literature about solo garlic or solo black garlic. But I read about multi-clove black garlic that higher total phenolic content can produce higher also of antibacterial activity.
Okay, thank you.
Yeah, thank you for the explanations. Still, there is a question from Dr. Ahmad Fatoni from LIPI, coming delivered to Mrs. Elvita. Why do you use the stem bark of mahogany for a source of endophytic fungi for antioxidant activity? It was based on the chemical structure found or other reasons? Yeah, please, Mrs. Elvita, if you have any comment or answer.
Thank you. In the previous study, we have isolated endophytic fungi from the parts of plant, such as leaf stalk, stem bark, root bark, and I have 11 a total 11 fungi. After the identified by morphological, one fungal from the leaf stalk and one fungal from the stem bark have a high similarity, and identification by molecular method, the fungal is as Cladosporium tenusinum. Both of the fungal have two spot the same of in TLC. Both of fungal the same of pattern spot in TLC, and one compound from
one component from the fungal is sorted from the lifestyle in active antioxidant, and the compound from the some fungus have a good antioxidant activity. The structure determine the structure based on the analysis of NMR, proton and carbon, based on the multiplicity, chemical shift, constant coupling, integration. The compound have the group of functional hydroxy. The compound is have aromatic compound with functional group hydroxyl, methoxyl, methyl ketone, and acetyl acid group. The compound from the standard and the compound from isolate the same of the lifestyle. Thank you.
Okay, thank you very much to Dr. For the explanations. Is there any comment from Dr. Abdi?
Thank you.
Yeah, Dr.
Thank you, moderator. Thank you, Dr. Vita.
Yeah, thank you, Dr. Vita. Yeah, for the presenters or then if you have a difficulty to explain in English, you may also use Bahasa instead of English. We are very welcome to host you. Yeah, I think this is the end of questions. There's no more question in this chat room, but we still have a little bit time. So if you still have any comment or maybe questions to the presenters, or maybe just comment to this section, just please, you're very welcome. And I also would like to let you know, please don't forget to fill in the registration attendance form. Oh, this is, I think if there is no questions, so we already coming to the end of this section. Before close these sessions, I would like to inform that please you may able to still joining these next coming sessions in around 15 p.m. Next coming session, and then in the closing seminar session as well. So again, we would like to say thank you very much for all presenters today. We have very good presenters all coming in this session. All 8 presenters master in the topics. So give a big applause to all the presenters. Yeah, we hope that from the session or from this seminar symposium, we get other perspective of our knowledge and our research. And the more important thing is how to gather the collaborations between us, each other, not only in internal our institution, but also external institution in Indonesia. And there are so many interesting topic as well as challenging study and dissemination for our research topic. Okay, thank you very much, and I am Andi Febri Santosa. Say thank you to all the participants who are already attending the sessions. See you. Bye bye to the
Halo.
Oke, Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. Welcome to this session for Parallel A. In this session, we will have three kinds of topic. The first one is pharmaceutical chemistry, pharmaceutical technology, and drug delivery system. In this session, we will have 6 speakers, and every speaker will present their topic for 10 minutes. There are some rules in this session. The first one is please have a stable internet connection. Second one is make sure that your computer has working camera and audio device. The third one is the chat room is only available for delivering questions to the ongoing discussion in the session. If you want to request to move your breakout for the presentation.
In this condition. Hello everyone. I would like to say thank you for coming in my presentation. It's a great pleasure to be here. It's a good symposium and I will not contribute also a little bit to the symposium. Let me introduce myself. My name is Nur Abdullah. I'm from University of Indonesia, and today I'm going to talk about the detailed study of the adalah HSDG formation as a biomarker of cancer risk due to malarial and common prevalent exposure group tentang reactions. This is the outline, and then for the introduction, based on the WHO data in 2019, cancer is the second leading cause of death globally. Many factors initiate the formations of cancer cells. You know that cancer is a very complex disease which is associated with a wide variety of risk factors. One of the that cause cancer is lifestyle factors, from the cigarette, diet, sun exposure, environmental pollutant, and etc. The factors generated xenobiotics such as malondialdehyde and chromium hexavalent that can produce selected species that will interact with macromolecules such as DNA, protein, and lipid. And then most cases of new cancer are detected at an affected stage so that it's more difficult to cure. And then in these conditions, early detections and identifications of potential malignancies of cancer cells is needed. Considering the negative effect of the chromium hexavalent on malondialdehyde, the study aimed to conduct an in vitro study to detect and identify the formations of the HSDG as a cancer risk biomarker. And then this is the formations of the HSDG. HSDG as a biomarker risk cancer can form due to exposure to DNA bases by xenobiotics, where in the study, xenobiotics used were malondialdehyde compound and chromium hexavalent compound. Malondialdehyde as the xenobiotic can cells from the exogenous and endogenous. From the exogenous such as pollutant, sun exposure, smoking habit, and etc., while from the endogenous source such as unhealthy diet, stress conditions, and etc. And then the second xenobiotic that is in this study source from the chromium hexavalent, where the chromium circulations in the environmental. Chromium is a naturally occurring element found in the rock, animal, plant, and soil. And then chromium hexavalent naturally occurring in the groundwater. Chromium exists in multiple oxidation stages. of which the hexavalent chromium and trivalent chromium stage are most prevalent biologically. And then, when the presence of the hydrogen peroxide as the oxidative agent for the chromium hexavalent or another metal transitions, it will trigger the formations of reactive oxygen species, where an imbalance in the amount of reactive oxygen species, such as superoxide radical, with the biological ability to detoxify the amount of reactive intermediate, causing toxic effect that damage cell, lipid, protein, and DNA. So, the guanine is evaporated, which comes to oxidation with the HSDG as the end product. And then this is the experimental method. This study, the work scheme in this in vitro study was carried out by reacting to the oxygonosine with xenobiotic malondialdehyde and chromium hexavalent with the following reactions were divided into 3 group reactions. After the sample has been reacted, the sample is then incubated with variations in temperature, incubation time, and pH value. And then the incubation result were analyzed by HPLC instrument. And then this is the result. From this result, from this study, the HSDG standard calibration curve from the validation method of HPLC was linear with correlations coefficient approaching 1 and met the standard linearity criteria. Moreover, the limit of detections and limit of detections to detect the analyte was 11.03 ppb and 36.77 ppb respectively. And then from the chromatogram in the HPLC instrument, the HSDG standard detected in time retentions 5.883 minutes, and this is used as the reference to detect and to determine the sample, the analyte in the sample. And then this is the results from the effect of the xenobiotic malondialdehyde, chromium hexavalent, and hydrogen peroxide to the deoxygonosine from the HSDG. This graph exposed along toward the NAA HSDG formations. From this picture, the result of the analysis shows that exposure of malondialdehyde to the oxygonosine able to trigger the formations of the NAA HSDG in 3 and due to its synergistic effect with malondialdehyde exposure in 3 hours of incubation time, otherwise for 16 hours of incubation time. And then this is the results about the effect of penton-like reactions toward the NAA HSDG formations. So from these pictures, the results obtained in this in vitro study by the additions of hydrogen peroxide as oxidator agent in penton-like reactions HP at 7.4, 16 hours of incubation time can increase the formations of HSDG. This indicates that hydrogen peroxide has synergistic effect with the presence of chromium hexavalent as a catalyst in forming hydroxyradical compound. And then this is the effect of temperature, pH, and incubation time toward the NAA HSDG formations. From these pictures, you can see this study found that the concentrations of the NAA HSDG was detected at only temperature 60 degrees Celsius, whereas at temperature 37 degrees Celsius, the formations of the NAA HSDG was detected but not quantified. This graphic also showed that there was correlations between pH and incubation time in the NAA HSDG formations, and then the effect of pH also correlated with the oxidation reductions potential of chromium hexavalent. Well, for the conclusions, the reactions of 2-deoxyguanosine with malonylaldehyde has formed the DNA index HSPG higher than LOD. Thank you for listening. May all tools we have shall be beneficial for all of us. Thank you.
Okay, thank you for your nice presentation. We will continue to the next speaker. The next speaker is Ibu Teni Ernawati from Research Center for Chemistry, LIPI. So please, co-host, play the video.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon. Hello everyone. I am Teni Ernawati from Indonesia Institute of Science, LIPI. Thank you so much for having me. I am delighted to be here to present the result of our project. I am so happy to share it with all of you at the International Symposium of Applied Chemistry 2020. Next, I would like to share the PowerPoint. Please wait a moment. The title of our project research is Investigation of the Potential COVID-19 Inhibitor from Casealata and Dendroctandra using Computational Approach. My name is Teni Ernawati from Medicinal Chemistry Research Group, Research Center for Chemistry, Indonesian Institute of Science. Outline of the presentation that I will deliver this afternoon is as follows. Introduction, methods, result, and discussion, and conclusion. COVID-19 is a recent coronavirus outbreak caused by SARS-CoV-2 beginning in early December 2019 in Wuhan, China, raising global health problems. The SARS coronavirus might peptidize SARS-CoV-2 plays an essential role in the life cycle of the virus and is a primary target for the development of anti-SARS-CoV. Also, the viral three-immunoprotein like cysteine protease, BCL4, enzyme controls coronavirus replication and is essential for its life cycle. In this slide was shown structure of SARS-CoV-2 imaged by CDC and also templates SARS-CoV-2 CL4NN. The protein consists of 2 chains and we only use the A chain. The background of this research is the utilization of Indonesian natural product for herbal medicine materials. The plants used here are Casealata and Dendroctandra. Active compounds from Casealata are emodin, aloemodin, and canferol. And active compounds from Dendroctandra are quercitrin and quercetin. In this slide was described traditional medicine production process. The first, the plant raw material are sorted and washing reckoning. Then, preparation of starting material as a product specification. Furthermore, the simplicia will be extracted using an alcohol solvent, dried, and then standardized of extract. The related product will enter to standardized extract. The last is packaging. Isolation of the active compound was carried out after the extraction process. And the objective of this study is to predict the activity of 5 compounds such as emodin, aloemodin, canferol, quercetin, and quercitrin that we obtained from Casealata and Dendroctandra in the SARS-CoV active site protein. Molecular docking is a method which predicts the preferred relative orientation of one molecule or key when bound in active site of another molecule or log to form stable complex such that free energy of the overall system is minimized. The molecular docking in this study was carried out by Autodoc on 4.2. The 3D structure of the base compound were made by Cambron 12. the crystal structure of main protease of it from protein database with ID 6LU7 to GTB 3M3V and homology docking were used as enzyme models and processed by Autodoc 4.2. The homology modeling template using Swiss model, application of molecular visualization using USCV Chimera 1.9 and Autodoc 4.2. Rampage for testing Ramachandran plot. Result and discussion. Tabel 1 was shown the structure and amino acid found in the active site pocket of two main proteases, MPRO and 3CL-PRO. 6LU7 is the main protease found in COVID-19, which been structured and repositioned in protein database and can be accessed by the public as of early February 2020. The less free energy generated from the results of the inhibition of compound with the protein SARS-CoV-2, the more active the prediction of this compound according to the docking simulation theory. Likewise, the smaller the value of the inhibition, the faster the kinetic of the required inhibition of the compound gene of the SARS-CoV-2 protein. Figure 1 shows the docking score data for all active compound in the sample and the commercial antiviral compound. So that among the simulated chemical compound structure, Nelfinavir as the commercial antiviral drug had the lowest docking score, while for the sample compound, almost all the SARS-CoV protein used except 3M3V. Quercetin, which is an active compound contained in dendrochitosan, had the lowest docking score for SARS-CoV-2, the protein in 6LU7 and 3M3V. Clenferol as active compound from Cassialata had the lowest docking score, SARS-CoV-2 to GTB and 3CL4 model homology. Figure 2 was described that interaction of ligand binding of 6LU7 SARS-CoV-2 with A. quercetin, B. clenferol, C. aloimodine, D. nelfinavir, E. remdesivir, F. hydroxychloroquine, and 3M3V SARS-CoV-3CL4 with G. quercetin, H. clenferol, I. hydroxychloroquine. The green dots show hydrogen bond. The binding of hydrogen with cysteine and histidine plays an important role in the inhibition of the SARS-CoV-2 protein. The binding of hydrogen with cysteine and histidine plays an important role in the inhibition of SARS-CoV because cysteine and histidine residue are the main constituent of the SARS-CoV-2 receptor. In addition to the main residue that play an important role, there are related amino acid residues that are no less important in producing small inhibitory energy. So that the SARS-CoV-2 molecule are inhibited among others proline, glutamine, leucine, methionine, glycine, methionine, cysteine, asparagine, and arginine. From the result of docking simulation of chemical structure, it can be concluded that those that have consistent active prediction on all the SARS-CoV-2 protein tests are quercetin, clenferol, and aloimodine for chemical substituent from Cassialata and dendrochitosan compounds, as well as nelfinavir, hydroxychloroquine, and remdesivir for commercial antiviral compounds. Conclusion, the interaction of ligand binding with SARS-CoV-2 shows that the interaction of amino acid residue plays an important role in maintaining functional conformation. The mechanism of enzyme and ligand interaction in this study is useful for understanding the prospective mechanisms of interaction of SARS-CoV-2 and ligand. Two parameters of the virtual filtering simulation that can predict SARS-CoV-2 inhibitor active are Low free energy gives low kinetic inhibition value. Gives free energy resulting from the simulation where sound, perceptive, temporal, and alloy have lower free energy gives and low kinetic inhibition compared to other sample compounds. Neofenavir, lopinavir, and hydroxychloroquine as commercial antiviral shows good results as antiviral compared to other commercial compounds. Thank you for your attention. Hopefully, it can be useful and provide information about the molecular docking of the potential COVID-19 inhibitor from Kasialata and Dendrochiptandra.
Okay, thank you so much for your nice presentation, Ibu Teni Errawati. Next, we will continue with the next speaker is Ibu Anjar Windarsi from Research Division for Natural Product Technology LIPI. So co-host, please play the video.
Thank you for the time. Assalamualaikum warahmatullahi wabarakatuh. My name is Anjar Windarsi. I'm from Research Division for Natural Product Technology, Indonesian Institute of Science. On this occasion, I would like to present my research entitled The Use of Proton NMR Spectroscopy and Chemometrics of Pattern Recognition for Authentication of Curcuma santoriza Adulterated with Zingiber montanum. The background of this research is that Curcuma santoriza or temulawak has been widely used in food, cosmetics, and also traditional medicine products. And its rhizomes is known to have several pharmacological activities such as antioxidant, anti-inflammatory, antibacterial activities, hepatoprotective, cardioprotective, and anti-cancer activities. And because of its high demand, it is potential to be adulterated with other species, especially having lower price, and adulterations is often carried out by unethical players for economic reasons to meet the demand. And one of the species which is potential to be used as adulterant in Curcuma santoriza is Zingiber montanum, because proton NMR spectroscopy is a fingerprint method. It is fast analysis, highly reproducible, highly robust, and it can simultaneously detect the primary and secondary metabolites of samples. Combined with chemometrics of pattern recognitions or chemometrics of classifications, such as principal component analysis, partial least square discriminant analysis, orthogonal projections to latent structures discriminant analysis, it becomes a powerful method in metabolite fingerprinting for authentication of medicinal plants. The first step of this research is rhizomes collections, and identification was carried out. Rhizomes were sliced, dried, and ground into powder. The powdered rhizomes were made binary mixtures of Curcuma santoriza and Zingiber montanum with several concentrations of Zingiber montanum from 0 to 100%. The powders were extracted using NMR solvent, deuterated methanol, and phosphate sulfur in deuterium oxides pH6 containing 3-methylsilyl propionic acids for chemical sieve calibrations, and then vortex, sonicated, and centrifuged. The supernatant was taken for NMR measurements, and the spectra were analyzed using chemometrics of pattern recognitions. This is the results of proton NMR spectra analysis of Curcuma santoriza and Curcuma santoriza adulterated with Zingiber montanum with several adulterant concentrations. And it is is very difficult to detect or to state whether the sample is authentic or adulterated when we don't have any information before regarding the samples, because from the adulterant concentrations of 10%, 25%, and 40%, they still have similar appearance or similar spectral pattern with authentic curcuma xanthorrhiza. Therefore, the use of advanced statistical analysis is highly required, such as chemometrics of pattern recognition analysis. Chemometrics of the LSDA could classify between pure and adulterated curcuma xanthorrhiza. The samples of authentic curcuma xanthorrhiza, authentic Zingiber montanum, and adulterated curcuma xanthorrhiza with Zingiber montanum can be classified perfectly, and the PLSDA models was evaluated using R2X and R2Y values, which indicated the good of fit of the model. And the higher the values of R2X and R2Y, close to 1, the better the good of fit of the PLSDA models. And other parameter used for evaluating the validity of the models is Q2 value, which is correspond to the good of predictivity of the models. And the Q2 value more than 0.5 is considered to be good of predictivity of the model. And the PLSDA model was performed using 7 principal components. Other pattern recognition chemometrics analysis, namely OPLSDA, perfectly separated between authentic and adulterated samples of curcuma xanthorrhiza with Zingiber montanum. And OPLSDA performed better separation than PLSDA. OPLSDA was carried out using 2 principal components and 4 orthogonal components. The R2X and R2Y values indicated the good of fit of the OPLSDA models, and the Q2 values also indicated the good of predictivity of the models. Chemometrics pattern recognition such as PLSDA and OPLSDA needs to be validated to confirm the validity of the model. And permutation test is one of the validation methods which is commonly used to validate PLSDA and OPLSDA model. The model was evaluated using original and permutated values, and these results confirm the validity both of PLSDA and OPLSDA models. From the above results, it can be concluded that proton NMR spectroscopy and chemometrics pattern recognitions of PLSDA and OPLSDA were successfully used for authentication of curcuma xanthorrhiza from Zingiber montanum. And combination of proton NMR spectroscopy and chemometrics pattern recognitions is potential to be used for authentication of medicinal plant. This is the end of my presentation. Thank you very much for your kind attention.
Okay, thank you so much for your nice presentation. Next, we will continue for the next speaker, Ibu Siti Agustina from Ministry of Industry, Republic of Indonesia. So, co-host, please play the video.
Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, I would like to say thank you for coming my presentation. My presentation about the extraction of antioxidant from Chlorella vulgaris for cosmetic. Indonesia is potential source of microalgae, especially Chlorella. Nowadays, it has been used as supplement. In this research, we try to apply Chlorella for cosmetic. protein, carbohydrate, lipid, vitamin, and mineral. It's good for cosmetic. Synergy of antioxidant in chlorella from chlorophyll, carotenoid, carbohydrate, and protein. Chlorophyll as antioxidant and pigment, acid degradation, but it has multi-benefit for cosmetic. So we can make encapsulation. Process encapsulation need spray dryer and maltodextrin. Spray dryer have fast method, low process, and can produce variant size powder. Maltodextrin have low solubility in water, high viscosity, and relatively low process, able to protect the bioactive compound from degradation. Obtaining chlorophyll antioxidant from chlorella vulgaris and applied to cosmetic in the form of cream and lotion. Method of extract chlorella is centrifugation process. After that, maceration process, and next centrifugation process, the final spray dry process. We can get encapsulated chlorella vulgaris. Method of application in cosmetic, encapsulated chlorella vulgaris is to formulation cream or to formulation lotion. We can get cream contain extract chlorella vulgaris and lotion contain extract chlorella vulgaris. Result and discuss. Maceration method can keep the bioactive compound from heating because it was done without heating, only by stirring. The moisture content of all chlorella vulgaris powders were below 10%. The S content decreased because the addition of maltodextrin will increase of evaporation. The highest carbohydrate content was powder chlorella vulgaris 0.05%. The addition of maltodextrin increased the carbohydrate content because its component is carbohydrate. The protein content of chlorella vulgaris reached 42 until 58% protein per dry weight of biomass. The fat content was not detected on various concentrations of chlorella vulgaris. The morphology of the powder displays a spherical structure, spheric, dispersed, and has a smooth form. Homogeneous Lorentz distribution across all spherical particles with monodispersity of chlorophyll inside each sphere. The encapsulation could increase the stability of chlorophyll as an antioxidant. The antioxidant content was not detected on powders with concentration chlorella vulgaris 0.01 and 0.05%. The highest antioxidant content was 11.83 milligram vitamin C per 100 gram sample produced by powder chlorella vulgaris 0.1%. Microbial contamination of cream and lotion were below threshold regulation of the Republic of Indonesia Drug and Food Supervisory Agency concerning amendment to BPOM regulation. It concludes that the cream with composition above is safe to be used. Organoleptic observation, the cosmetics, cream and lotion were not having aroma or odor and the texture still smooth until 8 weeks. The cosmetics were relatively stable. Conclusion, the bioactive compound from chlorella vulgaris was extracted by the maceration method and then encapsulated by spray drying method. The base powder, based on its antioxidant capacity, was chlorella vulgaris 0.1%, which gave 11.83 mg vitamin C per 100 gram sample. The powders have a spherical structure, spread dispersely, and smooth form. The base powder, chlorella vulgaris, has applied to cosmetic as cream and lotion form. The antioxidant content in cosmetic type creams was 4.95 mg vitamin C per 100 gram with antioxidant activity 719.75 mg per milliliter. And antioxidant content in the type of lotion was 4.73 mg vitamin C per 100 gram sample with antioxidant activity 660 mg per milliliter. The cosmetics, cream, and lotion were not containing microbial contamination according to BPOM regulation, and the organoleptic analysis showed it stable for 8 weeks. Based on this research, chlorella vulgaris have antioxidant activity which have potential to be developed as active ingredient for various cosmetics. Thank you for your attention.
Mbak Faiza masih di mute suaranya.
Oh, sorry. Okay, thank you so much for your nice presentation. We will have the next speaker.
Development of protein-based therapy has significantly risen since early 1980s, and most of them are available in parenteral dosage form. But we know that oral delivery is much more convenient and easier to apply compared than parenteral dosage form. Unfortunately, natural characteristics of proteins are much more sensitive to gastric environment than small molecules. Therefore, we need to encapsulate the proteins to avoid the destructive effect of gastric acid. In this study, we used a PLGA, which is a copolymer that consisted of lactic acid and glycolic acids, and this copolymer has been approved by FDA for medical application. Also, in the study, we used proteins, which are BSA and lysozyme. Both of these proteins are commonly used as a protein model in nanoformulation study. And what makes this interesting is the different PI of these proteins are significant. The protein was added into the oily phase by dropwise addition, and then immediately we sonicate at amplitude 40% for 2 minutes. And then this mixture was added with surfactant, also dropwisely. And finally, the last mixture was added into a larger volume of surfactant and then stirred at 600 RPM overnight at room temperature. On the next day, we separate the particles and sweetening by centrifugation at the high speed. These are the analysis for our particles. First, hydrodynamic size and zeta potential using zetasizer, protein loading and protein release, which was analyzed with SDSH and completed with densitometry. And the protein loading was confirmed using FTIR, and particle particles was visualized using scanning electron microscope. In a study, we tested 4 formulas, which consists of PLGF as the copolymer, protein, trehalose as protein stabilizer, and sodium chloride as a surfactant. And what I want to show you here is that the less we use PLGA and chloroform as organic solvent, and the more we use aqueous phase and even surfactant, the smaller particle size we obtain. But here, actually, the results was not really satisfying. So we can only get particles in micro size, and the PDI was quite good. But actually, we expected it should be less than 4.5. As we know that the lower PDI represents more homogeneous particle size. So next, for the protein loading and release, we only use the best formula that we had. Here, we incubate the PSA-loaded PLGA particle in acidic and neutral condition. And after that, we centrifuge the samples, and from the supernet, here we get the release of BSA. As you can see here, in lane 6 and 7, the release BSA is quite minimum, and there are still a massive proteins remain in the PLGA particles. Here, some serious concentration of PSA standard to quantify our proteins, but unfortunately and surprisingly, lysozyme was not encapsulated at all into the PLGA particles. As you can see here, even at the release or the remind lysozyme, there are no bands of protein at all. And this experiment has been repeated 3 times, and still we get a similar result. So here for the protein loading confirmation, we only use the BSL-loaded PLGA particles. Here you can see that from the spectra of the PLGA particles, there are no trehalose and sodium chloride specific peaks, meaning that after washing, the trehalose and the sodium chloride was successfully removed from the PLGA particles. And the most important thing is here, in the BSA-loaded PLGA particles, we get 2 vibration peaks appear at pip number 1657, which represents asymmetric stretching vibration of adenine, and a vibration peak at 1546 for asymmetric stretching vibration of glutamine, which are specific vibration peaks for amino acids. And from this data, we can conclude that the PSA was successfully incorporated into the PLGA particles. And to visualize the particles, here you can see that actually our particle was not homogeneous. Here we can see some small particles, but here we can see some big particles. So that's why the question, our PDI is quite high. So there are some factors determining particle size. First, concentration of PLGA used in the formula, and then ratio volume of aqueous phase versus oily phase. But here I want to discuss more in what determines the efficiency of encapsulation. So here, solution pH versus protein PI. So here we can say that pH of the aqueous phase is the most important factor influencing encapsulation efficiency. Proteins are nonetheless ionized at pH near its isoelectric point. So that's why the proteins are less soluble in the aqueous phase compared to that of in organic phase. Therefore, the protein leakage into the aqueous phase during the evaporation of the organic phase is minimized, leading to more effective encapsulation. And here you can see that the difference value from PI/PSA compared to pH of aqueous phase is much smaller compared than the difference value of PI of lysozyme than pH of aqueous phase. So to sum up, the current method was suitable for BSA encapsulation and showed high stability in gastric environment condition, but for lysozyme or maybe for other proteins, some adjustment are still required. And I would like to thank you and Obat 2019 and also for Research Center for Biology and Biomaterial Indonesian Institute of Sciences for facilitating us in analysis with SAM and FTIR. And also I acknowledge Ms. Norisa for her assistance on redrawing the FTIR spectrum using origin software. And last but not least, for your attention, thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much for your nice presentation, Ibu Riona. Next, we will have our next speaker is Ibu Sylvia Yusri from Department of Chemical Engineering, Swiss German University. So please play the video, co-host.
Good afternoon, all the participants and the committees. My name is Sylvia Yusri. The UFA or unsaturated fatty acid is the fatty acid that has one or more double bond in the chain, and it has numerous benefits, especially for skin. But unfortunately, the fatty acid is commercially expensive because it was extracted from fish oil and vegetable oil such as olive oil. So it is important to find another source of fatty acid that has lower cost. Canola oil is one of the best candidates as the source of the fatty acid because based on the previous research, it is proved that canola oil contains high amount of UFA. It is about 85%. But we have the problem because the canola oil is heat sensitive and it is also prone to oxidation because the content of the double bond. So to overcome this problem, we need to do the encapsulation to the canola oil. In this research, we use the HPLC because the HPLC is one of the common materials for the encapsulating agent that usually used to protect the fatty acid from the oxidation. And the objective of this research is to obtain the best ratio of the HPLC and canola oil that has the highest encapsulation efficiency. And then we need to do the formulation of antioxidant-rich topical lotion with encapsulated canola oil as the main ingredients. For the first, we need to do the extraction of the canola oil by using the food press expeller machine to prevent the degradation or the oxidation of the canola oil. Here are the process. So first, we need to collect the canola kernels, and then it is continued with the extraction of the food press expeller machine. And after that, we obtain the crude canola oil. So after that, we need to remove the residue or the impurities of the sample by using the centrifuge. And then finally, we got the refined canola oil. And for this extraction, we got the yield of the canola oil is about 0.149. So it is is quite similar to the previous research by Ismaili and Indiana. For the omega content, we successfully extracted high amount of omega content for the samples. So it is about 90.80% of omega content. And if we compare this composition to the previous research, we obtain a higher amount of omega-3 and omega-6, but a lower amount of omega-9. And we continue with the encapsulation of the candle nut oil. So we use two materials, raw materials. So first one is HPMC as the primary and the maltodextrin as the secondary raw material. And the encapsulation was carried out in the low temperature as about 5 degrees Celsius. And for the formulation, we prepare 3 samples with different ratio of oil to water, and it is from 2 to 3, 1 to 1, and 3 to 2. And for the first analysis, we do the peroxide value to analyze the rate of oxidation of the oil. So as you can see in the figure, we know that the encapsulated candle nut oil has a lower peroxide value if we compare to the non-encapsulated candle nut oil. So based on this data, we can conclude that the raw material is successfully protect the oil from the oxidation. And if we compare the 3 samples, the sample B has the best formula to protect the candle nut oil from oxidation because it has a lower rate of the oxidation. And if we do the analysis of the encapsulation efficiency, the sample B also show the best result for the encapsulation efficiency. But percentage of the encapsulated candle nut oil in the formula is from 2 until 10%. So we did 2 analyses for the solution. The first one is the antioxidant activity. If you see in the figure, you can see that the sample A, B, and C has the best activity of the antioxidant if we compare to the blank sample without the addition of the candle nut oil. So we conclude that the content of the candle nut oil improves the activity of the antioxidant of the lotion. And based on the data, the best antioxidant activity was obtained for the sample C with the 2% of the candle nut oil. And then we do the sensory analysis by 30 panelists, and the parameters were tested in the test is texture, the spreadability, the oiliness, and fragrance, and the overall acceptance. And based on the data, we got that there is no significant difference between each sample. So for the conclusion, we conclude that the encapsulation process was successful. With the sample B with the ratio of 1 to 1, the core to the raw material ratio was the best formulation in encapsulating of the candle nut oil. And then the antioxidant activity of the product did not correspond with the increase of the encapsulated candle nut oil concentration. And there is no significant difference of the overall physical likeness between the product. And we also recommend to analyze the microstructure of the microcapsule. And then we recommend to use the different raw materials and the formulation for the lotion. And also use the different formulation of the body lotion with the encapsulated candle nut oil. So that's all of my presentation. Thank you for your attention.
Okay, thank you so much for your nice presentation, Ibu Sylvia Yusri. Now we will come to the discussion part. In this part, we will we have 2 questions here, and for the speaker, who have difficulty to explain your explanation in English, you could use bahasa to make it easier. So the first question is from Bapak Ahmad Fatoni, LIPI, to Ibu Nur Afdila. The question is, can all cancer cells be detected by using the biomarker DNA ADAC of OHDG due to the effect of MDA and chromium-4? I thought it should be chromium-6, right? Exposure. So please to Ms. Nur Afdila to answer the question.
Okay. Thank you for the question from Bapak Ahmad Fatoni. I tried to answer the question. HSDG is one of the biomarker as an early indicator of potential marker for the emergence of cancer cell. So HSDG is formed due to exposure to base our DNA base that in these occasions the DNA base that exposed by the xenobiotic is the 2-deoxyguanosine. Why the 2-deoxyguanosine? Because the 2-deoxyguanosine is the DNA base group of guanine that most vulnerable to attack by the other chemical compounds. And then in this research, the xenobiotic that I use to trigger the formations of the HSDG as biomarker is the malondialdehyde and chromium hexavalent. And then to trigger the cancer cell by the biomarker DNA HSDG also can do to by another xenobiotic or by the another chemicals compound, not only by the malondialdehyde or chromium hexavalent, but also from by another chemicals like the polychlorinate, polychlorinate, or another transition metal or heavy metal. Thank you.
Okay, thank you for your answer. Any comment from Bapak Ahmad Fatoni or any audience?
Thank you very much for your answer.
Okay, so it's enough, I think. So we could jump to another question from Ibu Melati from LIPI to Mrs. Siti Agustina. Could you please show how much the ratio between maltose and vulgaris extract? Did you do optimization for maltose vulgaris ratio? Spray dry use high temperature. Was it possible the high temperature degrade or destroy the extract? So please, Ms. Siti Agustina to answer the question.
Thank you for your question, Bu Melati. I want try answer. This research is use the chlorella vulgaris solution with variation concentration 0.01% and 0.5% and 0.1%, and blended with maltodextrin 100 milligram per liter. This research we not we already optimization process for with spray dryer for the microalgae. The optimization is the result is the good is for microalgae 100 milligram maltodextrin per liter. Maltodextrin 100 milligram per liter. So this research we use only maltodextrin 100 milligram per liter.
Okay. Any comment from Ibu Melati for the answer
Okay. Thank you. And for the second
For the second question. For the
about
second
the
question.
spray dryer using the
Okay.
high temperature.
Yes. Spray dryer with the high temperature, but is the microalgae we already research is not so high different. If the natural after we spray dryer. So maltodextrin we can use the 50 milligram per liter, 100 milligram per liter, and 150 milligram per liter. The best is the for microalgae with the spray dryer is 100 milligram per liter.
So the high temperature didn't affect the activity of the microalgae, right?
Yes, this have the effect, but is we why we use the maltodextrin and spray dryer because is the before already I know maltodextrin is cheap and spray dryer is very spray dryer we can variation particle and low cost and compare we use the encapsulation another capsule.
Okay, thank you for the answer.
Okay, so we could jump to the third question from Ibu Elvita Unsri to Ibu Teni Ernawati. The question is, from this research, what are the characteristics of the compound that have the best potential to act as COVID-19 inhibitors? Yes, Ibu Teni Ernawati, please answer the question.
Thank you for the question, Bu Elvita. I will try to answer the question. Up to now, screening of potential of inhibitor COVID-19 still research and still exploration. But for this study, we found the potential of inhibitor COVID-19, the found for the flavonoid compound, such as for these cases is chimeferol, quercetin, and aloamodine. The flavonoid compound is the best potential candidate for inhibitor COVID-19. Thank you.
Any comment from Ibu Elvita or from another audience?
Thank you.
Okay. Okay, so is there any question to the speakers today? Okay, I think it's enough for today, for this session. So we will have another closing ceremony. It will be greatly appreciated if you could join the closing ceremony at 16:45. So I think thank you for your participation today, and I hope this program could give you more beneficial knowledge in the future. So thank you again, and please, if you have time, you could join the closing ceremony. Okay, bye. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Good afternoon, ladies and gentlemen. We really appreciate for your participation until closing ceremony. Our honorable speakers, moderators, participants. Finally, we come to closing ceremony of the ISAC 2020. We ask the Director of Research Center for Chemistry to give a closing remarks. Dr. Reni Meliana, the time is yours.
Thank you, Marisa. Assalamualaikum and good afternoon, ladies and gentlemen. Alhamdulillah, I believe that during these 2 days, all of us have obtained a fruitful discussion in this 6th ISAC 2020, which main theme is natural medicine and nutraceutical. We have learned a lot from the keynote speaker delivered by amazing our keynote speaker, Profesor Gunawan Indrayanto, thank you. Dr. Ambara Rahmat Pradipta, Dr. Muhammad Iqbal Qadri, we meet again. Niles Nirmal, PhD, also Associate Professor Nurul Huda and Professor Dr. Nina Artanti. Once again, on behalf of Research Center for Chemistry, LIPI, and the committee, thank you so much for delivering such important material so that all of us can deepen our knowledge. Furthermore, I would like to thank to all presenters and participants who have joined this conference. I certain that every presenter have presented their own research. I sincerely hope that through your presentation and discussion afterwards, all of us can contribute for better research in the future, and we can create good collaboration among us. Finally, I would like thanks to all the committee members for all this hard work organizing this conference and ensuring that it processed smoothly. I would like also to apologize for any inconvenience that you might experience during this conference. Ladies and gentlemen, all the participants and honorable speakers, once again, I thank you for your participation and wish you all stay healthy. See you again in the next 7 ISAC 2021. Thank you, Marisa.
Thank you, Dr. Yeni, for the officially closing our symposium. Ladies and gentlemen, and all plenary speakers, it's better for us to open our video. Maybe the committee will take the last documentation for today. Okay, thank you for the open the video. Also, we'd like to say thank you for spending the time with us, for all participants and also plenary speakers. And also, at last but not least, we hope we can meet on the 7th International Symposium of Applied Chemistry 2021. I'm Marisa Angelina. As a master ceremony of the symposium, would like to apologize for the inconvenience during our symposium. Good afternoon, have a nice day, and see you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you all.
Bye. Thank you.